Hello my dear friends, welcome to Live Prep. Myself Dr. Gajapati Varma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on YouTube channel. But test series can be given only through the app. Now in this particular video, we are going through the revision of molecular basis of inheritance, but here we are going only up to this DNA replication. So we are dividing this particular chapter into two revision videos, so that in one part we are studying up to DNA replication. In the next part we will go from transcription to the end of the chapter. So this is what we are planning. And one more thing is, my suggestion is first watch the molecular basis of inheritance videos and then you just go with principles of inheritance and variation. So that's why my suggested sequence is first go through molecular basis of inheritance and then you just go through principles of inheritance and variation, so that some concepts will be very easy to understand when we are going through principles of inheritance and variation. That's why I am suggesting this particular sequence. And after completion of these particular genetics lectures, then you can go with biotechnology. So this is the standard sequence, so that you can easily understand all these things. Clear? And now we are directly going with molecular basis of inheritance. In this we are going with the first part, where we are completing up to DNA replication. So quickly I am going with the NCERT content. Here also we are going through NCERT content only. Please understand, this much is enough. But when we are specifically studying about DNA replication, in the NCERT they have given only the machinery, but for understanding of the NCERT sentences, I am interlinking some concepts. So there I am going slightly beyond NCERT, but that is also ultimately to understand NCERT sentence only. But at the remaining all the contents, it is only NCERT. That much is enough for NEET examination. Now let us quickly start with this. In the basic introduction of this particular chapter, they are saying that molecular basis of inheritance. What is inheritance? It is the process by which the characters are moving from one generation to next generation. That is called as inheritance. And for this inheritance, what is the molecular basis? At the molecular level, what are the molecules that are responsible for this particular inheritance? This is what we are studying in this chapter. So what are the molecules responsible for inheritance? They are the nucleic acids. In general, they are the nucleic acids. Only nucleic acids can act as genetic material. Proteins cannot act as genetic material. Lipids cannot act as genetic material. Carbohydrates also cannot act as genetic material. It is always the nucleic acids which can act as genetic material. So if they are the nucleic acids, then what are the nucleic acids that are acting as genetic material? This is important. So DNA can act as genetic material. RNA can also act as genetic material. But where? Can both of them act as genetic material in the same organism? No. In the same organism, both of them cannot act as genetic material. Then who will act as genetic material? In what type of organisms? This is very important. So anyhow, this basic information already I have covered under cell chapter also, but once again I am telling you here, so please go through this. So here DNA is acting as genetic material. Yes, if an organism contains DNA, definitely DNA will be the genetic material. Please understand this. If organism is having the DNA, in many of the cases that is also having RNA. If both of them are present, if DNA and RNA both of them are present in the same cell, then who will act as genetic material? It is only the DNA. Please understand all these things. If DNA is not present, then only RNA will act as genetic material. So please observe all these points. So now we will go through some very important information. So observe very carefully. DNA acts as genetic material in most of the organisms, they said in NCERT. But what are those most of the organisms? So all living organisms. Why I am saying all living organisms? Because as per the cell theory, if you are using that cell theory concept here, so to call something as a living organism, compulsorily it should have cell as the basic unit. Yes or no? So all the living organisms contain cell as basic structural and functional unit. Yes or no? So this is what the point that we already studied in the cell chapter. So in this way, all the living organisms which are having the cells, in those DNA is the genetic material. Very, very important point. So that's why all the living organisms, that means all the 5 kingdoms, that means all prokaryotes and eukaryotes, that means all unicellular and multicellular organisms. Any of these things they may use in examination. So all these are having DNA as a genetic material. This is what, and explanation for this very clearly I gave under the cell chapter. Even in the revision also I have explained, so you can go through that. And apart from this, anyhow, these are having DNA as genetic material. Along with this, some viruses are having DNA as a genetic material. Is there any example from NCRT? Directly they have not mentioned in NCRT, but if we are observing Hershey and Chase experiment, in the Hershey and Chase experiment, what did they prove? They proved that DNA is the genetic material. Where? This is also one more thing. Many students are making mistake. See, students know that DNA is the genetic material. Unequivocally, it was proved by Hershey and Chase. Correct. Up to that, every student know. But in which organism? When we ask this question, students will say that in E. coli. No, they did not prove in E. coli. They proved in Bacteriophage. What is that Bacteriophage? T2 Bacteriophage. This name T2 was not mentioned, but Bacteriophage was mentioned. So bacteriophages are the viruses that are infecting bacteria. Majority of these are DNA containing viruses, and in their experiment, Hershey and Chase used T2 bacteriophage, and in that experiment, they proved that this bacteriophage has DNA as the genetic material. So that's why that experiment example you need to link up here. So in this way, all the living organisms and some viruses have DNA as the genetic material. Then what about the RNA containing genetic material? RNA is also acting as genetic material, that too only in some viruses, not in any of the 5 kingdoms. Please remember this. Many students are thinking that bacteria are having RNA as genetic material. No, bacteria will come under prokaryotes. They will come under kingdom Monera, and they are having DNA as genetic material. So only few viruses. So among the viruses, some viruses have DNA as genetic material and some viruses have RNA as genetic material. In the NCRT, they have given 2 important examples. One is tobacco mosaic virus and other one is QB bacteriophage. Q beta. Sometimes they may give the full form Q beta bacteriophage or bacteriophage Q beta. In any way they can give. So please remember this. This is NCRT example. Not only this, if you add one more important example, that novel coronavirus NCOV2, novel coronavirus which is causing COVID-19, that is also example for RNA as a genetic material. That is also example for RNA containing RNA as genetic material example. Clear? This is what some important information that we need to understand. Clear? Next one. RNA. RNA can act as genetic material in some viruses. Not only this, RNA is also having some other functions, but please remember, whatever the RNA that is acting as a genetic material, that RNA is not having this. This RNA is a different type of RNA. So in this, RNA is having different forms, and one form is genetic material form. That's it. And now another form is RNA can act as a messenger. So what is that RNA is called as? Messenger RNA. RNA can act as an adapter. What is the meaning of adapter? Actually in the revision part. So in the next part we will study about these things. So adapter means what? Connector. So it is connecting two things. What it is connecting? So who observed this particular one? Who proposed about the concept of this adapter? So it is the Crick. In the NCRT we will study in this chapter only. Okay, but in the next revision lecture we will study. So Crick proposed the concept of existence of an adapter. What is the adapter? Some connector, linker. Some connector should be there. So what it should connect? It should connect the genetic code, that means codon, with the amino acid. So since tRNA is connecting codon on one side and amino acid on the other side, now who is called as adapter? tRNA. So tRNA is called as adapter. And what is this full form of T? This T is the transfer RNA. Why it is called as transfer RNA? It is transferring amino acids from the cytoplasm to the ribosomes wherever they are present. They may be present freely in the cytoplasm, or they may be attached to endoplasmic reticulum, or they may be attached to nuclear membrane, outer membrane of nucleus. Yes or no? So these things we have already studied in the cell chapter. Interlink all these points. Next one, structural role. So some RNA molecules are having structural role. They are making some structures. What are these structures? It is making the structures called ribosomes. Ribosomes. So the RNA that is involving in the formation of ribosomes is called as ribosomal RNA. So in this way, the RNA having structural role is ribosomal RNA. It is making structures called as ribosomes. Next one. So some RNA are having enzymatic role. Not all RNAs, some RNA molecules are having enzymatic role. If RNA acts as an enzyme, it is called as a ribozyme. What are the important examples from the NCRT? It is the 23S rRNA. This point I have already informed under cell chapter. In the cell chapter when we studied ribosomes, there I have very clearly explained you. So 23S rRNA present in 50S subunit of 70S ribosome has enzyme activity. Similarly, so 28S rRNA present in 60S subunit of 80S ribosome has enzymatic activity. And what is the name of enzyme activity? This also I have mentioned in that cell chapter. What is the name of enzyme activity? It is the peptidyl transferase. And this information we will definitely study in the MBA, molecular basis of inheritance, but we will study under the translation process. So in the next revision lecture part, so we will study definitely about this particular information. Clear? So this is one general basic information that they have mentioned in the basic introduction. And what is DNA replication? Making another copy of DNA is called as DNA replication. And what is transcription? Synthesis of RNA from the DNA is called as transcription. And then what is translation? Synthesis of protein from the mRNA is called as a translation. So these are some names that they have mentioned. Anyhow, regarding the genetic code and then lac operon concept, DNA fingerprinting, all the things we will study in this particular chapter. This is what they have mentioned in the basic introductory paragraph. And then they have given a small paragraph. There they have given the information regarding what is the name of organism, how many base pairs are present. This type of information they give. That information we will study after few minutes, because before directly going with that information, we need to go with the next paragraph. There they have given the heading of polynucleotide chain synthesis. Polynucleotide chain synthesis they have mentioned. So in that polynucleotide means what? Many nucleotides are there. If many nucleotides are attached to each other, what will be formed? Nucleic acid will be formed. In the biomolecules we studied. In the biomolecules, when we are studying about the tissue analysis, there we studied about filtrate, retentate we studied. Retentate is also called as acid insoluble pool or acid insoluble fraction. In that it is having 4 components: proteins, polysaccharides, nucleic acids and lipids. Lipids are not true macromolecules, they are not actually the polymers, but remaining 3 are polymers. In that we studied about the nucleic acids. Nucleic acids are the long chains and each unit is called as a nucleotide. We studied all this information. I don't want to spend more time on it. Directly I am giving you the linking part. That's it. So nucleic acids are nothing but polynucleotides. So that's why polynucleotide chain synthesis means what? Nucleic acid synthesis, DNA synthesis, RNA synthesis, that's it. But before directly going with this, first we need to understand the single unit. What is the single unit? Our monomer is called as nucleotide. Tide is having 3 components. What are these 3 components? So one nitrogenous base, one sugar and at least one phosphate. Please remember I am saying at least one phosphate. It can have 1 phosphate, it can have 2 phosphates, it can have 3 phosphates also. Please remember whatever it is. If single phosphates, the 2 phosphates, 3 phosphates, if any number of these are present along with this, then it will be called as nucleotide only. So that's why while naming we will go with some specific naming like nucleoside, monophosphate, diphosphate, triphosphate likewise. So please understand these points. Anyhow, first let us see about the nitrogenous base. So within the nitrogenous base again 2 types of bases we are observing. Some bases will have basic purine ring. So that's why they are called as a purines. So what are the examples of purines that are present in the nucleic acids? Adenine and guanine. So that's why adenine and guanine are considered as substituted purines, because they have the basic purine ring, and in that basic purine ring some groups were substituted. So that's why adenine and guanine are substituted purines. Adenine is indicated with A and guanine is indicated with G. Next one under the pyrimidines, and these molecules are having basic pyrimidine ring, and these are cytosine, uracil and thymine. Cytosine is indicated with C, uracil with U, thymine with T, and these are considered as substituted pyrimidines. And here the spelling of thymine is very important, because there is a vitamin, it is the vitamin B1 and its spelling is THIAMINE. THIAMINE. Please observe this spelling carefully. THIAMINE is the vitamin B1 or vitamin B1, anything you can pronounce. And here we are talking about THYMINE, thymine. Please observe carefully. This thymine is a pyrimidine. So spelling of this one is very important for examination. So you need to observe this particular point very carefully. Clear? Now come to the sugar. And overall now nucleic acids, if we observe 2 types of sugars we are observing. Some types of nucleic acids are having ribose sugar. That's why they are called as ribose containing nucleic acids. So ribonucleic acid. So RNA. So RNA is having the sugar called as ribose sugar. whereas other type of nucleic acids are having deoxyribose. In specific, if you see that deoxy position is from second carbon, that's why it is called a 2-deoxyribose. So deoxyribose sugar containing, that's why it is a DNA, deoxyribose nucleic acid or deoxyribonucleic acid. So RNA contains ribose sugar, DNA contains deoxyribose sugar. This is what we are saying. And then phosphate group H3PO4. This is what present. And some nucleic acids may have one phosphate, some nucleotide. In general, I am talking about nucleotide. So some nucleotides are having one phosphate, some are having two phosphates, some are having three phosphates. Accordingly, the name will be changed. Clear? This is what the basic information regarding this. So in this way, individual nitrogenous base, individual sugar, individual phosphate with structures, with clarity, we have studied actually in the regular classes. In the revision, I am not going into those individual structures here. So directly I am going with superficial information. Now what we need to study? Now we need to study about the chemical names of this. You don't need to remember all these chemical names, but if possible, try to remember this. But or else, at least for the thymine, you need to remember. Thymine has another name, it is called as a 5-methyl-uracil. Quickly, I am telling you the names. Adenine is 6-aminopurine, and guanine is 2-amino-6-oxypurine, and cytosine is 4-amino-2-oxypyrimidine, uracil is 2,4-deoxypyrimidine, and thymine is 5-methyl-2,4-deoxypyrimidine. So 2,4-deoxypyrimidine is uracil, so 5-methyl-uracil. So in this way, thymine can also be written as a 5-methyl-uracil with structures, with every explanation we have studied in the regular lecture with more details. But here this name is enough. But if you can't remember this, it's okay, leave it. But thymine, also called as 5-methyl-uracil, this is compulsory because given in NCERT. At least this basic information, try to remember this. Adenine, guanine, cytosine, these three are present in both DNA and RNA. But uracil is present only in RNA, not present in DNA. And thymine is present only in DNA, not present in RNA. This is some basic information that we need to remember. And now we are talking about some names. So individual nitrogenous bases name we know, and individual sugar name we know. But when nitrogenous base and sugar are combined, now they are making a molecule, and that molecule is called as a nucleoside. So different nucleosides are having different names. So we need to remember that. In the NCERT they gave all these names very clearly in the biomolecules chapter. So that's why you need to remember all these names. And even in this MBA also, in a summary also, not in the summary, here itself, in the main paragraph itself, they have summarized all these names, that's it. So you need to remember this. So in this way, nucleoside is having two units. What are these? Nitrogenous base and sugar. To this nucleoside, if a phosphate group is attached, then it will form nucleotide. So in this way, nucleotide is having three components. What are these? Nitrogenous base plus sugar plus phosphate. So now it is forming nucleotide. First we will see the names of nucleoside, then we will see the names of nucleotide. But first we will talk with respect to RNA. Why I am saying with respect to RNA? Because first we are talking about ribose sugar. So in this way, what are the nucleotides that are observed in the RNA that you can understand? Now we will see. Adenine we know. Adenine, if it is attached to ribose, now it will form adenosine. See, at the end, osine is coming. With respect to purines, osine will come at the end. With respect to pyrimidine, adenine will come at the end. Understand this. So you can easily remember this. So guanine plus ribose is guanosine because it is a purine. But here cytosine, please remember, osine is present here, but this is not nucleoside. Even though osine is present, this itself is a nitrogenous base. Adenosine, guanosine are nucleosides, but cytosine is a nitrogenous base. So since already osine is present here, you cannot write osine here. So scientists have changed the name. So cytosine plus ribose will give cytidine. Idine is coming. And then uracil plus ribose is uridine, because these two are with respect to pyrimidine type. Okay, these two are with respect to purines, these two are with respect to pyrimidine corresponding nucleosides. In this way, nucleosides are formed. To this PI means what? H3PO4, phosphoric acid. In specific, if you are talking, it is orthophosphoric acid. It is phosphoric acid. It is not phosphorus acid. This is also previous NEET examination question. So observe carefully. It is a phosphoric acid. So when this acid is attached, now what happens in the name? In the name of all these things, ileic acids will come. Ileic acids will come. So observe this carefully. Adenosine plus phosphoric acid, adenylic acid. So these names we are giving with respect to single phosphate. With respect to single phosphate, observe carefully. And guanosine plus phosphate is guanylic acid. Cytidine plus phosphate is cytidylic acid. And uridine plus phosphate is uridylic acid. So in this way, corresponding nucleotides are formed. And these things here, single phosphate is there. So You can also write it in a different way. Adenylate acid, you can also write it as adenosine monophosphate, because on the adenosine one phosphate is there. Adenosine monophosphate is nothing but adenylate acid. Guanosine monophosphate is guanylic acid. Cytidine monophosphate is cytidylic acid. Uridine monophosphate is uridylic acid. So in this way, the names of nucleotide can be written with respect to how many phosphates that are present. So in this way, if single phosphate is present, monophosphates. If two phosphates are present, diphosphates. If three are present, triphosphate. Maximum three only. In this way, we are writing the names. And since we are talking for the RNA, here we are not talking with respect to thymine, because thymine is not present in RNA. Understood? If you understand these names, exactly these names you will study for DNA also. But in the case of DNA, before this name you need to add deoxy, 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 deoxy. But the major difference is, in the case of DNA, uridine will not come, thymidine will come, because uracil is not present in DNA, thymine is present in DNA. This is another change that you need to understand. So understand these names first. So as it is names, you can apply here also. Here also we just see it. Adenine is there, guanine is there, cytosine is there. Yes, as it is we studied. Adenosine, guanosine, cytosine as it is are there. But in before to this we are adding 2' deoxy, 2' deoxy, 2' deoxy. In the name also, adenylic acid, guanylic acid, cytidylic acid as it is names. But before this you need to add 2' deoxy, 2' deoxy, 2' deoxy. That's it. And anyhow here uracil is not there, thymine is there. Accordingly it will become thymidine or 2' deoxythymidine. And here it will become thymidylic acid or 2' deoxythymidylic acid. That's it. Simple. If you understand with respect to RNA, you can easily remember the names with respect to DNA. That's it. But here there is an important concept. What is the concept? Understand the concept here. If you are talking with respect to individual sugar, you are talking only for with individual sugar, then you should not use the prime. Then you need to say it is 2-deoxyribose. In the ribose sugar, at the second carbon, there is a loss of oxygen if you are comparing the original ribose sugar. So in the original ribose sugar, at the second carbon, OH group is present. But here instead of OH, remove the O, only H is present. This is what we are observing here. So if you are talking individual sugar, you need to write 2-deoxyribose. But when you are writing the names of nucleoside and nucleotide, you cannot write 2-deoxy. You need to write 2' deoxy. Why you are making 2'? Here you are saying 2-deoxy, but why you are saying 2' here? The concept is, here you are talking individual sugar, so no problem. So you are giving the numbers 1, 2, 3, 4, 5 likewise. But when you are talking nucleoside and nucleotide, nucleoside means nitrogenous base is present and then sugar is present. Here also nitrogenous base, sugar and phosphate are there. That means in these two cases, both nitrogenous base and sugar are present. Nitrogenous base is also having some carbons. So there is a numbering 1, 2, 3, 4, 5, 6, 7. And here also, according to the condition, right? If it is the purine, up to the number 9 we are going. If it is pyrimidine, up to number 6 we are going. And here along with this, sugar is also there. In the sugar again, 5 carbons are there. So there is a confusion between the numbering of nitrogenous bases and the sugar. So that's why in the nucleosides and tides, to avoid the confusion, what we are doing? For the nitrogenous bases, we are giving numbers 1, 2, 3, 4, 5, 6 for pyrimidine, 7, 8, 9, up to 9 we are giving for purine. And in the sugar, what we are doing? Instead of giving 1, 2, 3, 4, 5, we are giving 1', 2', 3', 4', 5'. So primes we are adding. Simply the original numbers are like that. At the top, we are adding the prime. And this priming will differentiate the numbering for the carbons present within the nitrogenous base and between the nitrogenous base and the sugar. In this way, we are avoiding this. So here, how exactly you are talking the second carbon of the sugar? It is the 2'. Here also it is the 2'. So that's why in the names of nucleoside and nucleotide, we are getting the primes. But when you are talking individual sugar, you are writing 2-deoxyribose. And this is the logical, very important concept you need to understand to solve some of the assertion and reason questions. That's why I'm spending time on it. Understand this point. Don't try to by heart this. Understand why you are adding primes here, why you are not adding prime here. So the logic you need to understand. So in this way, this complete table is done. Clear? In this way, nucleotides we are talking. Now here I have given an example of a nucleotide where there is one nitrogenous base, one sugar and 3 phosphates I have given. But names I will tell you, so observe carefully. Suppose it is the adenine. It is the adenine. Suppose it is the ribose sugar. Suppose you are talking only up to this. Adenine plus ribose is adenosine. And then if you are talking up to only single phosphate, adenine plus ribose plus single phosphate. Now what is the name? Adenosine monophosphate you can say, or you can say adenylic acid. Anything you can say. Suppose up to 2 phosphates you are saying. Now better don't use the adenylic acid term. We will go with specificity. Adenine plus ribose is adenosine with 2 phosphates. So adenosine diphosphate. And then if you are talking with 3 phosphates, adenosine triphosphate. In this way names we are writing. So please understand these names. In this way, instead of adenine, if guanine comes, what is the name? If cytosine comes, what is the name? If uracil comes, what is the name? So in all these cases, ribose is there. Suppose you are talking with respect to DNA, then instead of ribose you need to add deoxyribose, and then you cannot use uracil here, then you need to use thymine here. So in this way names. So this is what we have studied till now. These are the various names. And now we are talking with respect to the bonds. What are the various bonds that are present? Very clearly mentioned in NCERT. Observe carefully. Between nitrogenous base and sugar. Between nitrogenous base and sugar, any nitrogenous base, any sugar, no problem. So what are present here? It is simply the N glycosidic bond. Here I have given very specifically with respect to purine. If purine is there, N9 C1 prime. If it is pyrimidine, it will be the N1 C1 prime. In this way it will be attached, but anyhow you don't need those details. At least remember it is N glycosidic bond. Why it is N glycosidic bond? Naming also you need to understand. Why the bond between nitrogenous base and sugar is called N glycosidic bond? First I will tell you why it is called glycosidic bond. What is glycosidic bond? In the biomolecules very clearly we have studied. If sugar molecule is involved in the bonding, if sugar molecule is involved in the bonding, then that bond is called as glycosidic bond. Whether it is a ribose or deoxyribose, whatever it is, that is a sugar. Since sugar is involved in the bonding, now this bond is called as glycosidic bond. Correct. Up to this we understood. But why it is N glycosidic bond? Why it is N glycosidic bond? Why it is not O glycosidic bond? In this way different glycosidic bonds are there. So why it is N glycosidic bond? Because this sugar is attached to nitrogen N of nitrogenous base. Since it is attached to N nitrogen of nitrogenous base, it is considered as N glycosidic bond. So very clearly observe the NCERT words. You need to know all these things. Okay. So that's why it is called as N glycosidic bond. Very good. Next one. So fifth carbon of the sugar is attached to the OH group of the phosphate. And here due to the loss of water molecule, here there is a bond that is formed. And what is this particular bond is called as? This bond is called as, it is called as phosphoester bond. It is called as phosphoester bond. Normally in the case of normal ester, organic esters, what we are observing? Between the acid and the alcohol, this ester bond is formed. But here it is phosphoric acid. Since it is the phosphoric acid, anyhow this is the OH group alcohol, and here also ester is there. But which ester? Phosphoester is there. Because it is the phosphoric acid, we call this as a phosphoester bond. So you need to know the basic chemistry also. Then only you can understand all these basic things. Okay, next one. So in this way, here is the N glycosidic bond, here is the phosphoester bond. Very good. And then with this, what single phosphate is there? If another phosphate is attached to this existing phosphate, now what happens? Again water molecule will be there. So phosphoric acid, between two acids it is simply called as anhydride bond. Since phosphoric acids are involved, it is called a phosphoanhydride bond. And here also one more phosphate is attached, so one more phosphoanhydride bond. So in this way, if it is a nucleoside monophosphate, up to this if you are talking, what are the bonds present? One N glycosidic bond, one phosphoester bond are present, that's it. If you are talking about nucleoside diphosphate, up to 2 phosphates you are talking, then what are present? One N glycosidic bond, one phosphoester bond, and one phosphoanhydride bond are present. Suppose you are talking about nucleoside triphosphate, then how many bonds are present? One N glycosidic bond, one phosphoester bond, and 2 phosphoanhydride bonds are present. Bond counting is also important. Sometimes they will ask you, they will give you a so and so length of DNA or RNA, and in this how many so and so bonds are present. You need to be able to calculate this. So you need to know this basic information. So in this way, if you know the information for one unit, and then after this we will study about how exactly these molecules are attached, that also we will study now. So here I am explaining the same thing. So because of this you will understand the complete molecule. Once you know the complete molecule, then you can solve any type of question. So that's why you need to understand this basic information. So this is what. And in this example, this is the information for any nucleotide in general. But here specifically we are talking about adenosine triphosphate, adenosine, adenine plus ribose, adenosine having 3 phosphates is adenosine triphosphate. It is a nucleic acid. That is the energy currency of the cell. In the cell chapter under the mitochondria we study, ATP is called as the energy currency of the cell. This is the ATP structure. It is the energy currency of the cell. And in this energy currency of the cell, what are the bonds that are actually donating energy? These are the anhydride bonds. These are the phospho-anhydride bonds that are donating energy. In general, this ATP will be hydrolyzed to ADP plus Pi. When this bond is broken, this Pi is released and this molecule is ADP. That's why we will write the reaction as ATP gives rise to ADP plus Pi. This is the Pi, inorganic phosphate. Yes or no? So this is what we are writing. So when this bond is broken, it will release the energy. And very rarely it can be hydrolyzed to AMP when this bond is broken. AMP, adenosine monophosphate plus, this is the PPI that is released, and this PPI is further hydrolyzed. So ultimately what happens? 2 individual PI molecules will be released. So AMP plus 2 PI. In this case what happens? 2 high energy bonds are broken. So energy, double energy is released. Energy equal to 2 ATPs is released. Please remember, when we are doing energy calculations, one phosphoanhydride bond energy, it is approximately 30.5 kilojoules per mole, and this energy is called as 1 ATP energy. If 2 such bonds are broken, then while calculating energy, we call it as 2 ATP energy equivalents is released. In this way we are doing, but this information is a little bit higher for you, but understand these things. So this is what the information, because this information, energies and all these things, again you can link up with respiration chapter in plant physiology. Yes or no? There also we are studying many times regarding this. So you need to understand all this. So this is what about the basic structure, and please remember, the energy currency of the cell is a nucleotide. It is a nucleotide, that is nothing but ATP. Please remember this. This is the basic information. Before proceeding further, I will give another important interlinking information. When we studied about this chemical coordination and integration chapter, even in the revision also I have covered that point. At the end we studied about mechanism of hormone action. In the mechanism of hormone action, we studied about 2 types of mechanisms. One is for the peptide hormones, where they are hydrophilic, they cannot cross the plasma membrane, so they are binding to plasma membrane receptors. Yes or no? Other type are hydrophobic steroid hormones, they can directly enter into the cell. And there, first type, understand carefully, peptide hormones, hydrophilic, they cannot enter into the cell directly. That's why they are binding to plasma membrane receptors. So interaction of hormone and with receptor is creating hormone receptor complex that in turn generates second messenger inside the cell. What are the various examples of second messenger we studied? CAMP. What is that CAMP? Cyclic adenosine monophosphate. It is a nucleotide. CAMP, cyclic adenosine monophosphate, is a nucleotide. And even the not mentioned in NCERT, I gave another example. CGMP. What is that CGMP? Cyclic guanosine monophosphate, that is also a nucleotide. And along with this, CA plus 2, IP3, DAG, different other we studied. So forget about this. Focus on the first 2. CAMP, CGMP are second messengers. They are nucleotides only. They are also nucleotides. So cyclic AMP and cyclic GMP are the nucleotides which are acting as second messengers. In this way also they can ask an examination. This is the interlinking concept. You need to understand that is from the NCERT. You cannot say that it is out of NCERT, right? So understand this. So in this way there are some interlinking points are there. So that point also you need to understand. Now another interlinking point. Already I have mentioned when we studied about excretory production and their elimination. Even in the revision also I have very clearly mentioned. What is that point? Urea, uric acid, ammonia. Based on the excretion of these 3 nitrogenous wastes, we have classified the organisms. Ammonia means NH3 we are writing. Urea means NH2CO, NH2 we are writing. And the third one is uric acid. What is that? 2,6,8-trioxypurine. That is a purine. So that day I told you very clearly, right? So what is the point we have studied? Adenine and guanine are the nucleotides, or adenine and guanine are the nitrogenous bases. In simple we will talk. Adenine and guanine are the nitrogenous bases which are ultimately converted into a nitrogenous waste called as uric acid. So what are the molecules that are forming this uric acid? It is the purines called adenine and guanine. This point also you need to remember. These are all interlinked informations. If you don't interlink these informations, there is no use in preparation for NEET examination. Very clearly I am telling you. From the beginning itself I am telling you that don't go out of NCERT, but don't leave any not even a single word from the NCERT. In this way our preparation should be done. So with this all the interlinking points are completed. Now let us continue further. So till now we studied about the individual nucleotide structure, and in general we have studied about nucleoside triphosphate structure. And now these nucleoside triphosphates are used for polynucleotide chain synthesis. Many nucleotides, individual nucleotide we studied. Now we need to attach many such nucleotides so that polynucleotide will be formed. That polynucleotide will be either DNA or RNA based on the type of nucleotide. If the individual nucleotides, whatever they are coming, are ribosugar containing nucleotides, then the molecule is called as RNA. If the individual nucleotides are deoxyribosugar containing, then the molecule is called as DNA. But the mechanism is 100% same. What is the name of enzyme that is required? For RNA synthesis, RNA polymerase is required. For DNA synthesis, DNA polymerase is required. Enzyme is different. And nucleotides, ribosugar containing, deoxyribosugar containing. In this way, two types. So these are the differences, but the mechanism of the attachment of nucleotides is 100% exactly same. If you know for one, the same is for the other one. So anything you can take and we can understand. But before starting, and this one whatever I am reading, this was actually given under the DNA replication process, and this information is same everywhere for DNA replication transcription. And since we are studying polynucleotide chain here, so this is very important. What they are saying here, these nucleoside triphosphates, 3-phosphate containing nucleotides, these are having dual role. Dual means they are having two roles. What are these two roles? First one, they are acting as substrates because they themselves are attached to each other. That's why they are acting as substrates. And second one, they are acting as energy sources. They are acting as energy sources. They are acting as energy sources. But means what? They themselves have high energy phosphoanhydride bonds. Every triphosphate is having two high energy phosphoanhydride bonds. In this way, they are acting as energy sources. So they themselves undergo breakdown at the phosphoanhydride bonds. In this way, they themselves are providing energy. In this way, they are involved in making a polynucleotide chain. So in this way, these nucleoside triphosphates have a dual role. They are acting as substrates. They are acting as energy sources. These are basic points. Many students are not focusing on these small things. They are thinking that they understood everything. But when we are asking in the examination, they are making these small mistakes. Don't make any mistakes. At least NCERT content, please try to understand very clearly. Okay. Now we are going with the polynucleotide chain synthesis. Here we are talking about DNA synthesis. Mechanism is exactly same for RNA also. The only difference is in the DNA, here you observe carefully. Here I have written H here. If it is RNA, instead of H, OH will be there. That is the difference. Adenine, guanine, cytosine are present in both DNA and RNA. But RNA has uracil, DNA has thymine. That is the difference. But this attachment mechanism is 100% exactly same. So what you are saying here, suppose this is the first nucleotide. Please remember, for the synthesis always we need triphosphates. Mono and diphosphates are not useful. Mono and di should be converted into tri. Then only this tri can come directly for synthesis. So always we are talking about triphosphates only. Triphosphate should come. Suppose this is the first nucleotide is coming here. For DNA synthesis, what is the enzyme? DNA polymerase. It is holding the first nucleotide, and then it is receiving the second one. Now what happens is, for the first nucleotide, 3' OH means what? It is the third carbon of sugar. 3' OH. This OH is attacking on OH of first phosphate, means alpha phosphate of second nucleotide. For the second nucleotide, again 3 phosphates are there, right? So for OH of first nucleotide is attacking on OH of alpha phosphate of second nucleotide. So that what happens? Water molecule is removed. And during this process, the bond between this first and second phosphates is broken. So that now it is pyrophosphate. Combinedly, it is called as pyrophosphate. PPI, not single PI. It is PPI, inorganic pyrophosphate. And this pyrophosphate is further hydrolyzed. It is broken. So these two are released. But understand, this is high energy phosphoanhydride bond. This is also high energy phosphoanhydride bond. That means during this bond formation, two high energy bonds should be broken. So that energy is used here for making the bond. So please observe carefully. So these two phosphates are removed, and here OH, water molecule is removed, and this O is directly connected to this phosphate like this. O is directly connected to phosphate. So in this way, what happened? These two are attached. If two nucleotides are attached, it is called as a dinucleotide. In this way, two adjacent nucleotides are connected by, what are these bonds? Here this is one phosphoester, this is one phosphoester. Please remember, while writing what we are doing, between this carbon and this carbon we are writing. Please observe carefully, between this carbon and this carbon. What is this carbon? It is the 3' carbon. What is this carbon? It is the 5' carbon. So 3' to 5'. Direction is very important. I will focus on this point once again. 3' to 5'. What is the bond here? This is one phosphoester. Here is one phosphoester. So phosphoester plus phosphoester is phosphodiester. So in this way, between these two adjacent carbons, what is the bond that is present? Phosphodiester bond is present. That's why two nucleotides are connected by phosphodiester bond. So dinucleotide, in the dinucleotide, two nucleotides are connected by phosphodiester bond. This is the standard convention. You need to follow this. Understood or not? This is how they are saying. But while giving the ends for the DNA molecule, what you will say? This is the free end. The free end carbon is 5'. And in this way, sugar phosphate are only, please remember, only sugar and phosphate are involved in the attachment, backbone formation. But nitrogenous bases are not involved in backbone formation. They are projected at 90 degrees from the backbone. So backbone is formed by sugar and phosphate alternatively. And in this sugar and phosphate backbone, this is the first nucleotide. For the first nucleotide, 5' and carbon is free and it is having 3 phosphates. And in this way, if you are going for the last nucleotide, 3' OH will be free. So that's why for the first nucleotide, this end is called as 5' end. For the last nucleotide, this end is called as 3' end. So 5' end is having phosphate groups and 3' end is having hydroxyl OH group. This is what about. So DNA synthesis or RNA synthesis always occurs from 5' to 3'. This is what the meaning is. But if you are talking about phosphodiester bond formation, you need to talk that 3' to 5' phosphodiester bond. Once again I am repeating because many students are making mistake here. Complete DNA or RNA molecule, if you are talking, it is from 5' to 3'. It is based on free ends. But if you are talking about phosphodiester bond, what are the carbons involved? 3' carbon of first nucleotide and 5' carbon of second nucleotide. Understood or not? That's why it is called 3' to 5' phosphodiester bond. So these are some basic points. In this way, if many nucleotides are attached, it is called polynucleotide. If these are deoxyribosugars, this molecule is called as DNA. If these are ribosugars, this molecule is called as RNA. So mechanism is same. That's why in the NCERT, they did not give the heading as DNA synthesis or RNA synthesis. They gave as polynucleotide chain synthesis because it is a general term. For both of them, it is applicable. This is what. Understood or not? So in this way, so here, so since it is the deoxyribosugar, now this molecule will be DNA. So in this way, different diagrams are there. So you can go through. This is the NCERT diagram. So please observe this very carefully. But in this NCERT diagram, there is a mistake. What is that mistake? Observe carefully. In the NCERT diagram, so this is the DNA molecule. In general, they are saying that it is the DNA molecule. So now we will see this. This is the sugar molecule. For this sugar, now this is the second carbon. For the sugar, it is the second carbon. In general, if you are talking, it is 2' carbon. At the 2' carbon, you are making a line and leaving means what? In the chemistry, what you studied? So here, what should be there? Hydrogen should be there. So directly you need to write hydrogen here. If you are making a line, you need to mention as hydrogen. Suppose you are drawing a line and not mentioned hydrogen, simply you left it like this, like NCERT diagram. If you have left like this, what is the meaning of this? It is not hydrogen. If you are drawing a line and leaving there means carbon will come. Automatically the saturations with hydrogens will be done. Yes or no? The valency, the saturation with hydrogens will be done. So what it will be? It will become CH3 methyl group. Technically, as per chemistry, if you are using your knowledge here, you simply are making a line and leaving means here methyl group will come here. That is wrong. So please observe carefully. You should not draw line and you should not leave like this. This is actually mistake in NCERT. If you want to leave it like that, don't draw line at all. Then what happens? This is the carbon, and automatically 2 bonds were mentioned, means remaining 2 will be hydrogens only. That is correct. But if you want to draw a line, then you need to mention hydrogen compulsorily. You should not leave like this. Understood or not? So there is a small mistake, technical mistake. This is technically a mistake. So please observe this one. But anyhow, they have given an NCERT, so observe carefully. That's it. Okay. In this way, this is one strand. Same way, other strand is also synthesized, but in opposite direction. Because for this strand it is 3 prime and this is 5 prime. But this one it is opposite. So observe carefully. So when this strand is synthesized, it is synthesized from 5 prime to 3 prime. When this strand is synthesized, it is synthesized from 5 prime to 3 prime, because always DNA or RNA synthesis occurs only from 5 prime to 3 prime. It can never happen from 3 prime to 5 prime. This is very important point you need to understand. Anyhow, these hydrogen bonds and all, slowly we will study. Okay. This is what. So polynucleotide chain synthesis is always from 5 prime to 3 prime, whereas phosphodiester bond formation is from 3 prime to 5 prime. Very important concept. Many students are making a mistake in these. That's why I am spending more time on it, so that you will understand it very clearly. Next one, regarding the hydrogen bonds. Please remember, these hydrogen bonds we are studying between opposite strands. We are not studying within the same strand. Observe carefully. Within the same strand means these two. Opposite strand means these two. So where we are studying these hydrogen bonds? Between two strands. This is one strand, this is another strand. Between the nitrogenous bases of opposite strands we are studying this. Observe carefully. Between adenine and thymine of opposite strands, 2 hydrogen bonds. Between guanine and cytosine of opposite strands, 3 hydrogen bonds are present. In the examination, sometimes they may give a statement that 2 hydrogen bonds are present between adenine and thymine of same strand they will give. Many students don't read same strand. They will read that yes, 2 hydrogen bonds present between adenine and thymine, so it is correct. No, read the complete sentence. Of same strand if they give, then it is wrong. It is between opposite strands. So observe these sentences very carefully. Anyhow, I told you this. With respect to complete DNA or RNA, if you are observing, the direction is 5 prime to 3 prime. Only the phosphodiester bond, if you are observing, it is from 3 prime to 5 prime. Clear? Anyhow, more explanation with clarity I have explained in the regular classes, but here we cannot draw all the diagrams. That's what. So here 2 hydrogen bonds are present between adenine and thymine of opposite strands. 3 hydrogen bonds are present between guanine and cytosine of opposite strands. And here some students have the doubt. Do you need to remember what are the ones that are involved in the bonding? No. At the level of class 11th and 12th, you don't need to remember what are the positions that are involved in the bonding. No need. Simply remember how many. That's it. And this one just for understanding purpose only I have given. You don't need to remember what is the group, where it is exactly present. This information is not at all needed. I am very clearly telling you. Clear? Yes. And now we are going with this particular information. So generally for a particular molecule, for a particular organism, so whether DNA or RNA is acting as a genetic material. So whatever it is, so DNA or RNA length. See, generally what are the units for the length? Millimeter, centimeter, micrometer, nanometer, kilometer, meter. Likewise we are using the units. But generally for the nucleic acid like DNA and RNA, the length we are not representing in the standard units like millimeter, centimeter, like that we are not representing. Instead of representing like this, we are representing in the form of nucleotides or base pairs. When we will use nucleotide, if it is having only one strand, then we are using this as nucleotide. If 2 strands are present, then only we are using the term called as a base pair. What is this base pair concept here? Observe carefully. Suppose only one strand is there, 1 nucleotide, 2 nucleotide, 3 nucleotide, 4 nucleotide. So you can say 4 nucleotide. But here what happens, this is one strand, this is another strand. I will take this example only for explanation. So here 4 are there, here 4 are there. So instead of saying 4 plus 4, 8 nucleotides, instead of saying 8 nucleotides, what we say is, see, this one and this one are having the bonding. So now these two combined they will make one pair. So this is one pair, one pair, one pair, one pair. So how many pairs are present? 4 base pairs are present. So 4 base pairs means ultimately pair means what? 2. So 4 into 2, 8 only will come. But whether 8 are present linearly or 8 present as pairs, which one is exactly present? So this information also can be included if you are writing nucleotide or base pair. So please remember, we need to follow standard only. So if 2 strands are present, anyhow they are paired. So for this type of information, the standard way of representation is represent in the form of base pair. So for this what you will say? 4 base pairs. For this example, we will say 4 base pair. Suppose this strand is not there, only one strand is there. In one strand, you cannot say pairs. Then you need to count individual. How many nucleotides? 4 nucleotides. In this way, we are representing the length of DNA. But why you are representing like this? Can you convert this nucleotide number or base pairs into regular units of millimeter, micrometer, meter? Yes, you can convert. Simply by multiplication with distance between two nucleotides. Yes or no? Anyhow, that also we will study. So, since you are able to convert this base pair or nucleotide concept into regular units of millimeter, micrometer, yes, you can use in the form of these nucleotides or base pairs. So, in the NCERT already they gave these values. So, based on the units you just understand, nucleotide they gave, that means in this organism single stranded nucleic acid is there, but here they gave base pairs, that means double stranded is there. But whether it is linear or circular, based on this you cannot say. So, based on analysis only we can understand. But anyhow, this particular information was not given in NCERT, but I have given here for your understanding purpose. So, observe carefully. Bacteriophage. What is bacteriophage? Bacteriophages are nothing but viruses. These are the viruses that are infecting bacteria. So, here 2 examples they give. One is bacteriophage 5x174. It is 5x174. And another bacteriophage is lambda bacteriophage lambda. So, these are the 2 examples they gave for viruses. And Escherichia coli is a bacterium. One example for bacterium they gave, and the other example is eukaryote human they gave directly. For the human they gave haploid condition and diploid condition. So, in these conditions, so how many base pairs or how many nucleotides are present, that we will see. Bacteriophage 5x174 has 5386 nucleotides. Since in the units they gave nucleotides, it is never having base pairs because it is a single stranded. But what type of single stranded? Circular. And what is the genetic material? DNA. Please remember, even though these two are viruses, both of them are having DNA as genetic material. Please remember this. Some viruses have DNA as genetic material. No? These are the 2 examples from the NCERT. You need to remember this. Anyhow, T2 bacteriophage of Hershey-Chase experiment, that is also there, and these 2 are also there. Add this. You need to remember this. So, all the examples of these NCERT are having DNA only. So, you are not studying RNA example in this table. So, it is DNA. Understood? Clear? Since these are base pairs, these are all double stranded. Since nucleotides, it is single stranded. Up to this, it is okay. But whether it is circular or linear, that you need to understand, right? Now, observe carefully. For these things, for the bacteriophage 5x174, it is circular, whereas bacteriophage lambda is having linear double stranded DNA. Okay, this is having circular single stranded DNA, this is having linear double stranded DNA. And E. coli bacterium, whenever we are drawing the bacterium diagram, we are making the circular only, double stranded circular only. So, you can understand circular double stranded DNA. And human chromosomes are there, that means linear is present, so linear double stranded DNA. So, in this way logically you just try to understand. For these 3 you can remember, but for these 2 you need to by heart whether it is circular or linear, okay, because you did not study anything regarding these viruses. So, at least this information you just try to by heart if possible. But anyhow, this information was not given, but I suggest you to remember that. Clear? Okay, nice. And now after this in the NCERT actually directly they have gone for that Chargaff's rules and Wilkins and Franklin's contribution and then Watson-Crick model, but I am going through the timeline. So, these experiments were done later on. So, before to this actually there are some experiments for the search for genetic material. So, first we will complete all these 3 experiments, then we will go with that Watson-Crick model and all. Clear? The experiments are very simple. I will quickly try to complete these things so that we can proceed very quickly further. Search for genetic material. Here in the NCERT they have given a paragraph. There they have given the contributions of various scientists like Sutton and Bowery. So, what is the contribution of the Mendel? Okay, and what are the contributions of some other scientists? Likewise, they have given a big list. So, please go through that basic information, that is a simple information. And even those various different scientists have contributed so much things, but ultimately, no one knows exactly what is the genetic material. This is what the conclusion of that paragraph. Even the different scientists discovered so many things. So, no one has clearly said that this is what the genetic material is. So, this is what the problem is. So, in the search of that genetic material, there are some experiments, and here we are first studying about the Griffith's experiment. After the Griffith's experiment, we will go with Avery-MacLeod-McCarty's experiment. After this, we will go with Hershey-Chase experiment. So, under the search for genetic material, 3 experiments are there. First, we are studying about the Griffith's transforming principle. So here we are studying about two important things. One is transformation, other one is transduction. Transformation and transduction. Please observe carefully. Griffith experiment and Avery-MacLeod experiment. These two are based on transformation, bacterial transformation. But Hershey-Chase experiment is transduction experiment. I will tell you what is the meaning of this. What happens in transformation? Bacterium receives DNA from another bacterium or from the outside source, so that it receives another DNA molecule, so that now it is undergoing some changes. Once again I am repeating, in the transformation what happens is a bacterium receives DNA from surrounding medium or from another bacterium, so that some of the properties of this bacterium will be changed. That's what happening in the transformation. But what happens in the transduction? In the transduction, a bacterium receives DNA from virus. This is the difference. Please remember. So that's why in this experiment you are talking only about the bacteria. In Avery-MacLeod also we are talking about bacteria only. But in the case of Hershey-Chase experiment, we will talk about a virus and a bacterium, because that is a transduction experiment. Understand the meanings of these experiments so that it will become very easy for you. And here in the Griffith's transforming principle, anyhow we need to talk about bacterium. And here what is the name of the bacterium? Streptococcus bacterium. Strepto means it is like a chain of coccus. Coccus means what? Spherical shaped bacterium. So in this round shaped bacteria, they are present one after the other like a chain. So that type. And why the name pneumonia was given? It is not pneumonia, it is pneumonia, species name. Pneumonia, at the end E was given. So name was given as pneumonia because it is causing disease called pneumonia. Understood or not? Since this bacterium is able to cause pneumonia, it is called as pneumococcus. It is called as a pneumococcus bacteria. Clear? And this streptococcus pneumonia information was given in the Griffith's experiment. But in the Griffith's experiment, this bacterium is infecting the host called mice. Yes or no? Plural mice, singular mouse. Singular. If you are talking with respect to one, it is called mouse, M-O-U-S-E, mouse. If many you are talking, M-I-C, mice we are using. Many students are not knowing what is the difference between mouse and mice. Mouse is a singular, one animal, and mice are many, plural. That's what the difference is. But another important interlinking concept is, in human health and diseases also they mentioned this name. Streptococcus pneumoniae and Haemophilus influenzae infect the alveoli of lungs, and then they are causing the pneumonia. Yes or no? So please understand, the Streptococcus pneumoniae can infect human and also can infect mice. Understood or not? And it can cause the disease called as a pneumonia. If it is not treated, it can kill the individual also. That's why in this case, when a specific strain is given, mice will die because it is causing a deadly disease called pneumonia. Understood or not? This is what the important point is. And in this particular bacterium, there are two important strains. One is called as R strain, other one is called as S strain. What is the R strain? Rough nature. It is making the rough colonies because it is not having mucus coat around the cell wall. Since there is no mucus coat, it is having a rough appearance and it is making rough colonies. And since mucus coat is not there, actually mucus coat is responsible for disease called pneumonia. Since mucus coat is not there, R strain cannot cause the disease. So even though R strain is given to the mice, they will survive. There is no problem to them. But what about the S strain? Smooth. Why is it smooth? Around the rough cell wall, it is having a smooth mucus or polysaccharide or mucopolysaccharide coat. Yes or no? So because of this, it can cause the disease pneumonia, and that's why when S strain is given to the mice will die. This is what the information we need to understand before starting the experiment. That's what they are saying. Clear? In the experiment, actually, 4 setups are there. What is the first one? So in the first one, one group. Please remember, there are mice of different groups. To one group, live means living R strain was given. Anyhow, since though it is living, R strain cannot cause disease. So even though R strain was given, mice did not die. They are still surviving. That's what he observed. And that too, from the lungs of this particular mice, he recovered living R. Anyhow, they are not causing disease, and he injected R and he recovered R from the lungs. So no problem. That is as expected only. Next one. So next one, he has given the living or the live S strain he has given. But S strain is virulent strain. It is pathogenic. It can cause the disease, right? So that's why when this was given, mice died. And that too, from the lungs he recovered live S strain. So given S strain, recovered S strain. So this is also expected, and they died, that is also expected. So no, it is not something new. But here what he did, heat killed S strain. What he did, S strain previously it was given in a live form. But here what he did, before injecting to mice, he has heated it. By exposing to heat, what happens? Bacteria will die. So that means dead bacterium was given here. Here in this case what he did, dead bacterium was given to the mice. Already before injection the bacterium was dead. Can dead bacterium cause the disease? No, it cannot cause the disease. So that's why they are surviving. And anyhow here from the lungs, S bacterium was not required because already they are dead. If they are already dead, how you can grow them on the plate? You cannot grow them. So that's why he did not recover S strain here. So up to here everything is as expected. But the last one is important. What he did? He mixed living R strain and dead S strain. But technically if you see, individually they cannot cause the disease. So what is expected? Mice should survive because individually when they were given, there is no damage to the mice. Yes or no? But when they were combined and this combined mixture was given to the mice, then what happened? He observed that mice were dead. He observed the dead mice. And second important point is, he gave the live R strain, so from the lungs he recovered R strain live, that is good. But he gave heat killed S. Even though heat killed S were given, from the lungs he recovered live S. How this is possible? He gave heat killed, but he recovered living S. How this is possible? Do you think that the dead bacterium again became live? No, it is not like this. Then what is the conclusion he gave? So for that conclusion, conclusion is important. How Griffith has concluded this? Please remember here, Griffith don't know what is the exact material that caused the death. He don't know. But what he proposed? But something, definitely something from the heat killed S strain. Something from heat killed S strain entered into live R strain. Something from heat killed S strain entered into live R strain, so that live R strain slowly transformed into live S strain. And since live S strain was formed, it can cause the disease and it can kill the mice. That's why the mice were killed, and that's why the live S strain were obtained from the lungs. This is what the conclusion he gave. Once again I am telling you the conclusion. So what Griffith concluded? Something, he don't know that something. And what that something is called? That something is nothing but a transforming principle, but he don't know what exactly it is. So something from heat killed S is entering into live R strain, so that live R is converted into live S. So since it became live S, it can cause the disease, so it killed the mice. This is what the explanation he gave, and that is correct. Explanation is correct, but the thing is, he don't know what is that something. Simply he called it as transforming principle, because that particular thing, something, transformed normal R strain into pathogenic S strain. So physical transformation has happened. So it has transformed R into S, and so he called it as transforming principle. That's it. But he don't know the biochemical nature of that particular molecule. So in this way, experiment is related to genetic material. That something that is moving is definitely genetic material, but he don't know what it is. So in this way, that is the first experiment related to search for genetic material, and that experiment was further continued by Avery-MacLeod and McCarty. And the most important point is, these are the scientists, they first proved that very clearly. I am telling you, please observe carefully. First proved that DNA is the genetic material. Who first proved that DNA is the genetic material? One option is Griffith, other option is Avery-MacLeod-McCarty, next option is Hershey-Chase. Many students are writing Hershey-Chase. Don't write that. Who first proved that DNA is the genetic material? It is Avery-MacLeod-McCarty. Understood or not? And when you will write Hershey and Chase, you will write Hershey and Chase only when they ask that unequivocal proof that DNA is genetic material. That word unequivocal proof should be there. If that is not there, you should not go for Hershey-Chase, because actually these scientists have very clearly proved that DNA is the genetic material. Even though some biologists were not convinced, if they are not convinced, how we are responsible? So, but these scientists have proved it very clearly. So, who first proved that DNA is the genetic material? It is Avery-MacLeod-McCarty. 90% students are making mistake here. If they ask that who unequivocally proved that DNA is the genetic material, then only you should write Hershey-Chase. Very clearly in the regular classes I have explained this. So, please remember this. Don't make any mistake here. Clear? So in this way they proved it. And here also what is the experimental organism? Again the same bacterium Streptococcus pneumonia. Again the same R strain and S strain only. Same things they have used. So Griffith's experiment is a transformation experiment. So Avery-MacLeod-McCarty experiment is also a transformation experiment. So this is the point that you need to understand. But here in this experiment, actually in the internet I did not get both the diagrams. I got only one diagram. So I will try to explain here. So what is the last step of Griffith's experiment? Heat killed S strain. Heat killed S strain and live R strain were mixed. Then this mixture was given to the mice and the mice died. This is what the last and most important step. And in that case, what was proposed by the Griffith? Something from heat killed S strain is entering into live R strain so that it is converted into live S strain. Please remember, some students are thinking that all the R will be converted into S. No, that is never possible. All the bacteria, all the R strain cannot convert into S. Out of these many R, some of the R will be converted into S strain. That's it. So majority will be R only, but some will be S strain, and that some S are enough. They can cause the decision, they can kill the mice. Okay, please remember this. Don't go for any wrong interpretations. Many students are doing wrong interpretations in the genetics. That's why they are making many mistakes. Don't interpret any wrong things on your own. If you are having any doubts, ask the faculty, so we will tell you. Clear? Yes, this is what the last step of Griffith's experiment, and this is where this Avery-MacLeod-McCarty experiment will begin. What Griffith said? Something from heat killed S strain. So what is that something? So these scientists have isolated or purified major macromolecules. What are the major macromolecules here? Carbohydrates, lipids, and then proteins, DNA, RNA. These are the major macromolecules that are present in this. So definitely one of these should be entering into this one. So that's why what they did, they took the R strain culture. Okay. So in the first step, what they did, from the heat killed S strain, carbohydrates were isolated and they were added to R strain. And here what they obtained, R strain. And here what they observed, everywhere R strain will be there. Observe carefully, because even though there is a transformation, entire R cannot be transformed. So definitely some R will be there. So along with the R, where actually they have observed S. See this. When carbohydrates from heat killed S were added to R, and here they observed R only. That means there is no S formation. When lipids from heat killed S were added to R, here also they observed R only. So here also there is no S. When proteins from heat killed S added to R strain, here also they observed only R strain, there is no S strain. When DNA from heat killed S added to this along with R, they also observed S. Observe this carefully. But when RNA from heat killed S added to R strain, here they observed only R. Only R. So what is the meaning of this? What is the meaning of this? The meaning of this experiment is when DNA from heat killed S added to R strain, it resulted in the formation of S along with R strains. So anyhow, our focus is not R strain, our focus is S strain. So focus on the S strain. So when the S strain was formed, when the DNA from heat killed S was added to R strain, then the S strain was formed. That means what? That something from heat killed S is going is nothing but DNA. So DNA is the genetic material that is moving from heat killed S strain into live R strain, so that some of the R strain got converted into S strain. Sorry, extremely sorry. They got converted into live S strain. This is what the conclusion is. So they clearly proved that it is the DNA that is moving. Not only this, they did another experiment. So for this actually I got diagram from the internet. Now let us see this. And this is what the diagram is. Second experiment. What is this another experiment? This is actually the mixture. I will tell you this one also. And what is this mixture? So from the heat killed S strain, they got carbohydrates. Simply I am writing all this: carbohydrates, lipids, proteins, RNA and DNA. They got all these individual things, and now they made a mixture of this. This is the mix. Understood or not? So now what they did, this mix C plus L plus P plus R plus D, this is the mix, right? So here also, because it will be easy for you to understand. That's why I am writing all this. This is the mixer, and this mixer they made into different samples. They made into different samples. So in this way, 5 samples. Let us see. This is what the mix of all these things, and all these were obtained from heat kill S strain. This is what. And then what they did is, instead of directly adding to the R strain, before adding, what they did, they have treated with different enzymes. What are these enzymes? Here they have treated with carbohydrases, and here they have added lipases in a separate tube. All these things in separate tubes. Here they have added proteases, and here they have added RNAs, and here they have added DNAs. Understood or not? First they did this. First they have taken the mixture and then they have treated with different enzymes in a separate tubes. Then what happens? Carbohydrates will break down carbohydrate, but DNA is normal. Lipases will break down lipids, but DNA is normal. Proteases will break down proteins, but DNA is normal. RNAs will break down RNA, but DNA is normal. But DNAs will break down DNA. DNA is broken. So please understand this. Now what they did? Now this they have added to tube having R strain, and then everywhere it is the R strain. Okay, separate samples. Let us see. And what did they observe? Everywhere definitely R will be there. Everywhere definitely R will be there. Observe carefully, because if there is a transformation, entire R cannot transform, right? R will be there. Now where you will see the S, observe carefully. Here carbohydrates will break down carbohydrates, but DNA is normal. DNA of heat killed S is present, so it can transform R into S. Yes or no? Next one, lipase will break down lipids, but DNA is normal, so this DNA can transform R into S. Next one, protease will break down proteins, DNA is normal, so this DNA can transform R into S. Next one, RNAs will break down RNA, but DNA is normal, so this DNA can transform R into S. But when DNase is used, it is directly digesting the DNA only. When the DNA of heat killed S is directly digested, now it cannot transform R into S. Since there is no DNA, intact DNA is not present, it is broken. So since it is broken, it cannot transform R into S. So in this case, you are getting R only. So this is also clearly saying that it is the requirement of DNA. DNA is required. If this DNA is broken, there is no formation of a strain. That means DNA is compulsory. So it is the DNA from heat killed S strain that is entering into the R strain, so that some of the R strain are converted into S strain. So in this way, that Griffith's transforming principle is nothing but DNA. So in this way, they proved that DNA is the genetic material. So this is what they experimented. Different enzymes they have used. So that particular component is digested. But in some cases, DNA is normal. But in this case, DNA is used. So DNA is not normal. So here R strain only. But in all cases, R and S will be present. So from this experiment, it is concluded that DNA is the genetic material or hereditary material. In the NCERT wording, if you say genetic material, also called as hereditary material. So who first proved this? It was proved by Avery-MacLeod-McCarty. Very clear. But even though they proved like this, some biologists were not convinced. If they are not convinced, we cannot do anything. But these are the first scientists to prove that. So further experiment, in that experiment, they also got convinced. So that's why there you are using a word called unequivocal. So that we will study under Hershey-Chase. So before going with the Hershey-Chase experiment, in the NCERT they have asked that, can you write some difference between DNA, plural they gave, DNAs means what? Yes means either here this S indicates that it is a plural. So many DNA molecules. This is DNase, it is an enzyme. So what is the difference here? DNA means it is deoxyribonucleic acid, and it is the polymer of nucleotides, and it is acting as genetic material in majority of the organisms. But what is this DNase? ASE is an enzyme, and it is the deoxyribonuclease, and since it is an enzyme, it is the protein. Protein means it is the polymer of amino acids. It is not polymer of nucleotides, it is the polymer of amino acids. And what is the function of this? It will break the DNA into small fragments. That is the function of this one. So how it is breaking? It is breaking exactly the phosphodiester bonds. In this way, it is breaking the DNA into small fragments. These are the important differences between DNA and DNase. Clear? Very good. Next one. With this, the two experiments are completed. Now we are going with the third one. So what is the third one here? So Hershey and Chase experiment. What is the important word that we are using? Unequivocal. What you can easily understand the meaning of this. Vocal means what? Vocal sound. Equivocal means equal sound. What is the meaning of equal sound? Suppose I am saying something, you are also saying the same thing. So at the equal sound, we both of them are arguing. So in simple, I am trying to tell you the meaning. So equivocal is what? Argument. Unequivocal is no argument. That means there is no doubt on it. There is no further argument. There is no doubt. That means it is very clear. Unambiguous. Ambiguous means doubt. Unambiguous means no doubt. It is clear, no doubt, and it is not a questionable. In this way, unequivocal proof was given that DNA is the genetic material. By whom? By Hershey and Chase. But please remember, who first proved that DNA is genetic material? Avery-MacLeod and McCarty. Who unequivocally proved that DNA is the genetic material? Hershey and Chase. So that's why wordings are very, very important. So please observe them very carefully. But these are what type of experiments? Are they transformation experiments? No. These are transduction experiments because virus is involved here. From the virus, the DNA is going into the bacterium. So that's why it is transduction experiment. And please remember, in this experiment, what is the experimental organism? It is not E. coli. E. coli is not the experimental organism. It is the T2 Bacteriophage. The name T2 was not given in NCRT, but please remember, simply they give Bacteriophage. Technically, it is the T2 Bacteriophage. Sometimes they may give T even number, odd number, you know, no? Even. So 2, 4, 6, 8. These are the even numbers. So that's why they may give. If they give, they may give it as sometimes T even Bacteriophage. Like that also they can give. Still, it is correct. But technically, it is the T2 Bacteriophage. This is the experimental organism, and in this organism they are trying to know what is the genetic material. Why they are doing like this? Because even though Avery-MacLeod-McCarty proved that DNA is the genetic material, at that time many scientists, they thought that protein might be the genetic material. They thought that protein might be the, not protein is the, protein might be the genetic material. That's what their thinking is. But originally it is not. So these scientists have clearly proved that it is not the protein, it is only the DNA that is acting as genetic material. They proved it with the help of an experiment. So that experiment we are trying to understand here. So in this first, in the one paragraph, they are talking about what are bacteriophages actually. Bacteriophages are the viruses that are infecting the bacteria. Clear? So in this way they are propagating with the help of this particular bacteria. Bacteria means the host for that particular virus. So these viruses cannot divide on their own. For their division, compulsorily they need a host. So for the bacteriophages, the host is the bacterium. And here what we are studying is, we are studying about the T2 bacteriophage. And what is the host for T2 bacteriophage? E. coli bacterium. That's why we are using the E. coli bacterium for the growth of this particular virus. That's it. Clear? And here we need to understand some important points. The points are, in the case of this T2 bacteriophage has only two components. What are the two components that are present in this? So this is the protein coat. This is the protein coat. Okay, so it is having the protein coat like this, and inside this it is having the DNA. Please understand this. This T2 bacteriophage has only two components. Only two components. One is protein coat, other one is DNA molecule. So since it is having only two components, now what is the relation with the previous experiment? In the previous experiment of Avery-MacLeod-McCarty, in Streptococcus pneumoniae, they proved that DNA is the genetic material. Not in the T2 bacteriophage. In Streptococcus pneumoniae, DNA is the genetic material. That's what they proved. Nice. But what many scientists are still thinking, protein may be the genetic material. That's what they are thinking. So now both of them are present in this. So now what they did is, these scientists have taken this T2 bacteriophage, which is having both DNA and protein coat. Yes or no? When it is infecting E. coli, it is producing next generation viruses. Up to that it is fine. Up to that they know. But between these two, what is the exact genetic material? At that time they don't know. Today we know much information about all these things, but at that time they don't know. So now their aim is to know whether it is protein or the DNA. Which one is exactly acting as genetic material? That's what they wanted to know. And for this, they are trying to make two types of viruses. T2 bacteriophage only, with radio labeling, they wanted to make two things. What is the one? In one, they want to make S35. For the protein, we will say S35 labeled protein coat. in one thing. So in this particular virus, the DNA should be normal. It is the normal DNA. In this way, one type of virus where only protein coat is radio labeled, but DNA is not radio labeled. But in the other case, protein coat is normal. So this is the normal protein coat. It is the normal protein coat. That means radioactive labeling is not done here. But it should have radio labeled DNA. Understood or not? So here directly I am giving you the conclusion. For the protein coat, what was the radioisotope that they have used? S35 used. And here for DNA labeling they use P32. Here I don't want to go into more details inside because it will take so much of time. But in the regular lecture very clearly we studied this. Why hydrogen H3 tritium, right? Tritium or tritium. Why you cannot use that? With respect to carbon, if you are talking, C14 is also radioactive, but why you are not using that? With respect to oxygen, if you are talking, O18 is radioactive, but you are not using that. Why? With respect to nitrogen, N16 is radioactive, but you cannot use that. Why? These are some important points that I have discussed in the regular lecture. But please remember, I am giving the conclusion. If you are using H3 or N16 or O18, likewise, any of these radioisotopes if you are using, the problem is it can go into both protein coat and DNA. So these, whatever the list I said now, these can go into both protein coat and DNA, then there is no use. So you should select an element in such a way that it should be present in only one component. It should not present in other component. If you are talking about the sulfur, it is present only in the protein. It is not present in DNA. So you are using this. If you are talking about the phosphorus, it is present only in the DNA. It is not present in the protein coat. That's why you are using this. So in this way, it is called as a specific labeling. Specifically, only one is labeled so that other one is normal. So in this way, some viruses are like this, some viruses are like this, not all viruses. So in NCERT, very clearly they used some viruses, very clearly this. Some are like this, some are like this. Not that all of them are of same type. Understood or not? That's why in the experiment also, this type of viruses were used in this one. This were also used in other bacteria. E. coli only, but these two experiments were done simultaneously. Here also you are doing, here also you are doing, steps are same. That's why simultaneously you are studying this. So please understand each and every word of NCERT. Don't try to by heart this. Very good. Anyhow, in the DNA we studied about nitrogenous base sugar and phosphate. In that phosphate, this phosphorus is present. So this P32 will go directly into the DNA. Very good. But this S35, where it will go in the protein? Protein means amino acids. 20 different types of amino acids are there. Out of these 20 different amino acids, what are the amino acids which contain sulfur? Sulfur containing amino acids. In the biomolecule, very clearly I told this. What are the sulfur containing amino acids? They are cysteine and methionine. Sulfur containing amino acids are cysteine and methionine. These amino acids are present in the protein coat. That's why if S35 is used, it will go into the protein coat. In this way, specific labeling will occur. In this way, what happens? One set, and here protein coat is labeled, and here DNA is labeled. And now what they did? Now there are total 3 steps are there. What are the 3 steps here? First step is called infection. Second one is called as a blending. Third one is called as centrifugation. What happens in the infection? So these radio labeled viruses were taken and they are infected to their host. What is the host? E. coli. Is this E. coli radio labeled? No, it is not radio labeled. It is normal E. coli. You need to take normal E. coli. Please understand this. And when they are infecting, then what happens? Their genetic material DNA. Anyhow, now I am saying genetic material, but at that time they don't know the DNA is going into the cell. Here also DNA is going into the cell. Very good. Next step, what happens? After this, once DNA is going inside, this protein coat is present only on the outside and it should be removed. And how you are removing this? By blending. It is the vigorous shaking, vigorous mixing you are doing. So that what happens? This protein coat will be separated from surface of the E. coli bacterium. But in a mixer, it is a liquid, right? So uniformly they are distributed. Both virus and this protein coat of virus and bacteria are uniformly distributed. Now you need to segregate them. How you will segregate them? With the help of centrifugation. What is the major principle of the centrifugation? Based on the density, the molecules can be separated. If it is more denser, it will settle faster. If it is lighter, it will be present at the top. So in this way here, who is more denser? Bacterium is large in size, so its density is more, so it will come to the bottom of the tube. and above that there is a liquid. In that liquid this protein coats are floating. So now ultimately what you will get? You will get total 2 fractions. Each side, here you are getting 2 fractions. Suppose this is the tube. So here you are getting 2 fractions. Here you will get bacteria. Here you will get bacteria, and inside this bacteria viral DNA is present, and here is the fluid. This is called supernatant. Supernatant, it is a general term, supernatant. The fluid floating above is called supernatant. In this supernatant what is present? The protein coat of this virus is present. This is how they are present, and then they have measured the radioactivity. What they observed? What they have observed is, when protein coat of DNA was radio labeled, supernatant is radio labeled, but bacterial cells are not radioactive. Supernatant is radioactive, bacterial cells are not radioactive. What is the meaning of it? The protein coat of virus is not entering into bacterium. This is what the conclusion is. Here what they have observed, where DNA of virus is labeled, here what they observed, supernatant containing protein coat is not radioactive, but the bacterial cells are having radioactivity. What is the meaning of it? Viral DNA is entering into the bacterium. So what is the overall conclusion? Once again I am telling you, here protein coat of virus is not entering into bacterium. Viral DNA is entering into bacterium. So the one that is entering into the bacterium will be the genetic material. Understood or not? This is what the conclusion is. Further continuation also they did, and there they observed that in this case, wherever the viral DNA radioactivity present within the bacterial cells. So now when these cells are allowed for next generation, even then in the next generation also some viruses, not all, some viruses got radioactivity because this DNA as it is going into some viruses. So in this way they proved that it is the DNA that is acting as genetic material which is entering into the bacterium, and hence it is responsible for synthesis of new viruses. But protein coat is not entering into the bacterium. So protein is not the genetic material, DNA is the genetic material. Very clearly they have proved this. So that's why every biologist got convinced. That's why they used a term called as unequivocal proof that DNA is the genetic material, came from Hershey-Chase experiments. Clear? So who first proved that DNA is the genetic material or hereditary material? It is Avery-MacLeod-McCarty. But who unequivocally proved that DNA is the genetic material? It is Hershey-Chase. So please observe all these things. Don't make any mistakes. Understand the experiment. So what is the conclusion from this side? What is the conclusion from this side? What is the overall conclusion? Each and every point. Don't make any mistakes. Clear? Nice. So with that, this search for genetic material is completed. So they know that DNA is the genetic material. That's it. Very good. But even though they know that DNA is the genetic material, they don't know exact structure of DNA. So now we need to talk about structure of DNA. So for understanding of the structure of DNA, we need to first go through Chargaff's rules. Then we need to go through the contributions of Wilkins and Franklin. Then we are going with Watson-Crick double helical DNA model structure. Chargaff's rules. He analyzed double stranded DNA molecules. Upon analysis of double stranded DNA molecules, he concluded that amount of purines is equal to amount of pyrimidines, and amount of adenine is equal to amount of thymidine, amount of guanine is equal to amount of cytosine. And actually this information is very important for Watson and Crick model, and in that model they have proposed about complementary base pairing. For the proposal of complementary base pairing, Chargaff's rules are very important. Now I will tell you some questions. Observe and understand the question first, then you just give the answer. Who proposed complementary base pairing? Who proposed complementary base pairing for DNA? Is it Chargaff or Watson-Crick? Many students are making mistake here. Who proposed complementary base pairing? It is Watson-Crick. Watson-Crick proposed complementary base pairing. Understood or not? Chargaff did not propose. But what is the information related to Chargaff? Watson-Crick proposed complementary base pairing based on experiments of Chargaff. So Chargaff's rules are the basis. These are the basis for the proposal of complementary base pairing by the Watson and Crick. So don't make any mistakes here. Understand the contributions of each and every scientist and who proposed what. That also you need to observe carefully. Clear? This is what. And they also observed that equimolar proportion of sugar and phosphate are also present. And here this point was not there in NCERT. So base ratio, he even calculated the base ratio. What is technically base ratio? So for the numerical questions, this is very important. Sometimes they may give some data, they will ask you to calculate base ratio, that's it. They will not tell you what is that ratio exactly. This is the base ratio. A plus T by G plus C is called as base ratio. So this is the standard. For a particular species, this value will remain the same. Different species are having different values. Some standard examples are these. For E. coli it is 0.93, for human it is 1.52, but this information is not there in NCERT, so you don't need to remember this. You don't need to remember this. Just for understanding, I am giving like this. So for different species, these values are different. So in this way, Chargaff's rules are very important. Based on the Chargaff's rules only, actually, so Watson and Crick proposed the complementary base pair. Nice. Next one. Next one we are observing is the contribution of Wilkins and Franklin. And actually what they did is they have crystallized the DNA molecule. Please remember, it is not such an easy thing. So crystallization process will take so much of time, and that too, DNA is a very long polymer. So it is highly difficult to isolate DNA as it is first. Intact DNA, isolation of intact means as it is DNA, that is very difficult, first thing. And second thing is, even though you are isolating this, and converting it into a single crystal is highly difficult thing. That will take so much of time, and that needs so much of expertise also. So that's why it took so much of time to isolate that DNA intact and crystallize this. But finally these scientists got succeeded in that, and then once they got this single crystal, and then they have gone for this crystallography. So in the crystallography they have got some image like this, and then they have gone for the analysis of this particular thing. And please remember, in the Watson-Crick model, we are studying about there are two strands, they are coiled around each other, and then so and so in each helix these many base pairs are present, distance between these base pairs is so and so, and the diameter is so and so. Likewise, some statistical information we are studying now. Please remember, entire statistical information was given by these scientists only. So that's why, so Wilkins has received the Nobel Prize along with Watson and Crick. So they have contributed so much, okay. But they gave the basic information, but the model was proposed by Watson and Crick. But the basic statistical information was obtained from these scientists only. So to avoid the repetition, we are not studying here, directly we are studying under the model. But please understand, statistical information was given by Wilkins and Franklin. This is what the contribution of these scientists. And finally, we are studying about the most interesting model, that is the Watson and Crick DNA double helical model. And here we are talking the structure for bDNA. There is aDNA, bDNA, cDNA, ddNA, zDNA. Likewise, there are more than dozen forms are present. And here the standard DNA that we are studying is the bDNA, because normally in our physiological conditions in our cells, this is the DNA that is present. That's why we are studying about this. But remaining other forms we are observing in some experimental conditions. So we are not worried about that. So we are focusing only on the bDNA model. So in this model, who proposed this? Watson and Crick proposed. When they have proposed it in 1953, they published this research in the article journal that is called as Nature Journal. But anyhow, this point, Nature Journal name is not needed for you. So in this way, they have proposed this, and Watson and Crick along with Wilkins, they received Nobel Prize for their contribution here. Clear? So now we are talking about some important features of this DNA double helical structure here. So in this, what they propose, so two polynucleotide chains are present, two strands are present. They are the two polynucleotide chains are present. And how they are present? These are wound around each other. They are helically coiled around each other. This is how they are present, but actually they are coiled around an imaginary axis. And what direction of this coiling is? Right-handed coiling. Anyhow, separately I kept a diagram, there I will show you here. And in this particular one, if you are observing the each strand or each chain, so here there is a backbone. So who is forming the backbone? For the backbone, there are two questions are there. What are the molecules that will form backbone? It is the sugar and phosphate. Molecules, if they ask, you need to write sugar and phosphate. What is the bond that makes the backbone? If they are asking about the bond, you need to write phosphodiester bond. So which one exactly they are asking? Molecules, if they ask, phosphate and sugar alternatively. Bond, if they ask, it is the phosphodiester bond. So in this way, it is forming the backbone. Very good. And then from this backbone, nitrogenous bases are projected. And where they are projected? They are projected around 90 degrees. They are projected around 90 degrees. Please observe carefully. So since these are projected, suppose this is the backbone. So these nitrogenous bases are projected like this with 90 degrees. 90 degrees they may say, perpendicular they may say, or right angle also they may say. Any of these words can be used. So observe carefully. So in this way they are projected. Nice. Next one. Next one, they are saying that the two strands with respect to directions, 5' 3' directions, if you see, they are present in opposite direction. One is from 5' to 3' like this. one is from opposite direction. So since they are present in opposite directions, so it is anti-parallel to each other. This is with respect to polarity. With respect to polarity, they are anti-parallel to each other. This is what they are saying. So next one, they are saying that the reading of each of these strand is from 5 prime to 3 prime, because each strand is synthesized in 5 prime to 3 prime direction. And with respect to hydrogen bonds, if we see the nitrogenous bases of opposite strands, they are interacted with hydrogen bonds. So in this way, they are having complementary, they are making complementary base pairs. And how many hydrogen bonds are present? Between adenine and thymine, 2 hydrogen bonds are present, and these are from opposite strands. Between cytosine and guanine of opposite strands, how many hydrogen bonds are present? 3 hydrogen bonds are present. So in this way, hydrogen bonds are interlinking these 2 strands. Next important information is regarding the uniform distance. There is a uniform distance between 2 strands. And how this is possible? Because so if it is a purine always pairs with pyrimidine. So purine pyrimidine, purine pyrimidine, purine pyrimidine, purine pyrimidine, purine pyrimidine. So always you just see any pair, always one purine pairs with one pyrimidine. That's why almost the uniform distance is able to maintain. So that's why here there are 2 important points. Don't get confused, listen carefully. First one, the term anti-parallel we are using with respect to polarity, because one is 5 prime to 3 prime, opposite 5 prime to 3 prime. So with respect to polarity, anti-parallel, very good. But with respect to distance between 2 strands, suppose there are 2 lines, distance is always constant between these 2. Can you say parallel or not? Yes, you can say. Mathematically, these 2 lines are called parallel lines, because the distance, anywhere you see the distance, it is exactly same. So that's why read the sentences carefully. In the DNA molecule, the 2 strands are parallel with respect to distance between them, correct? In the DNA, the 2 strands are anti-parallel with respect to polarity, that is correct. So that's why please observe the sentences carefully. Parallel with respect to distance between them, anti-parallel with respect to polarity, correct. Now, another point is, suppose they have not mentioned anything. The 2 strands of DNA are, this is the question, the 2 strands of DNA are, one option is parallel, other option is anti-parallel. Then what you will do? In that case, you need to go for anti-parallel only, because if they are not mentioning anything, means default condition is with respect to polarity. So you need to go for anti-parallel only. Clear? Read the sentences, don't make any mistakes. Next one. Here, so this is the right-handed coiling fashion. This is how actually the two strands are coiled around a central imaginary axis, and this is the right-handed coiling. So this is how we are observing the DNA double helix. Okay, and one more, I will come back to this one, and now we will go with interlinking part. So when we studied about the biomolecules, I told you one interlinking part, the helical structure that we are observing in the starch, alpha helix that we are observing for the protein, collagen triple helical structure, and DNA double helical structure. All these are examples for secondary structures. This is one interlinking, very important point, and this one already I have covered under the biomolecules also. So please remember for examination, all these are from the NCERT. Remember this, okay? All these are secondary structures, okay? Now let us continue. Now we are talking about the pitch. What is this pitch means? The length of one helix is called as pitch. And in this pitch, or in one helix, one coil, one turn, any name they can use, all these are same only. So what is the length of this particular one? Very clearly they have mentioned that it is 34 angstroms. In the angstroms if you are saying 34, but in the nanometers if you are saying it is 3.4 nanometer. And in one coil, how many base pairs are present? 10 base pairs are present, and all these base pairs are present at a uniform distance. If they are present at uniform distance, what is the average distance between 2 adjacent nucleotides? It will be 34 divided by 10. So it will be 3.4 angstroms or 0.34 nanometers. This is the distance between 2 adjacent base pairs. That's why this value is very important for us. When you are converting the number of base pairs or nucleotides into micrometer, millimeter, nanometer, what you need to do? You need to multiply with this distance. You need to multiply with distance between 2 adjacent base pairs, that is 0.34 nanometer or 3.4 angstroms. Anyone you can take it. Okay, this is what we need to do it. So this is one important information we need to remember. Next one. What is the diameter for this particular DNA molecule? It is having 20 angstroms diameter. In the nanometer it is 2 nanometer diameter it is having. Clear? Next one. So these nitrogenous bases, they are not just present one above the other in a straight line. Actually, they are helically coiled and they are present as a spiral steps. So when they are present as spiral steps, definitely they are present one above the other, but not exactly above, slightly rotated. That means there is an angle between these two. So what is the angle between two adjacent base pairs? So for one complete helix, 360 degrees. And how many base pairs are present? 10 base pairs are present at equal distance and equal angle. So here what you will get? 360 divided by 10. So that will be 36 degrees. So the angle between two adjacent base pairs, or it is the helix raise, R-A-I-S-A, helix raise per base pair. So with angle, with respect to angle, if you are talking, it is 34 degrees. With respect to distance, if you are talking, it is 0.34 nanometers. Please observe this carefully. Okay. Next one. So finally, they are saying that base pair stacking. What is the base pair stacking? So they are present one above the other, right? Even though they are spirally coiled, they are present one above the other. So between the nitrogenous bases also hydrophobic, not hydrogen, hydrophobic interactions are there, and they are present one above the other. Presence of one above the other is called stacking, stack, right? So this base pair stacking is also very important along with hydrogen bonds present between the strands for stability of DNA molecule. This is what that they are saying here. Clear? So there are so many factors that are giving the stability for the DNA molecule. And here there is one very important point that you need to understand. What is the important question is, in the double helical DNA molecule, this is not, I am not talking about this one, I am asking one another important question, observe carefully. In the double helical DNA model, the important point we need to notice is phosphodiester bonds are present on outer sides, whereas the nitrogenous bases are present at the center. Nitrogenous bases are present inside the helix, whereas phosphodiester backbone is present on outer sides. Why is it like this? Assertion reason question, it is very important, observe carefully. The reason is DNA. You just talk about DNA in the prokaryote, eukaryote, wherever it is. In prokaryote, it is present in cytoplasm. In the eukaryote, it is present in nucleoplasm, mitochondrial matrix, chloroplast stroma. Yes or no? This is the location of DNA. And wherever it is, this DNA is surrounded by some fluid. What is that fluid? Cytoplasm, nucleoplasm, mitochondrial matrix, chloroplast stroma. This is the fluid, water, right? So surrounding the DNA, water is present. So observe the logic carefully. Phosphodiester backbone means sugar phosphate backbone is hydrophilic in nature. It can interact with water. That's why it is present on outer sides. Whereas the nitrogenous bases are hydrophobic in nature. They cannot interact with water. That's why they are present inside the double helix. This is also another very important logical assertion reason based information. So please remember this. So with this, the basic structure of DNA double helix is completed. Now we are going with packaging of DNA molecule. Why this packaging should occur? Now we will go with the basic calculation. So here they are talking about what is the length of DNA, what is the length of DNA in the meters, for the human diploid cell. In the human diploid cell, so what you are saying, in the human diploid cell, you know the number of base pairs, 6.6 into 10 to the power 9 base pairs, you know, but you need to convert this value into the meters. So how you will convert this? These many base pairs multiplied with distance between two adjacent base pairs. What is it? 0.34 nanometer. Multiply with this. Nano means what? 10 to the power minus 9, right? It is the 10 to the power minus 9. Sorry, there is a small mistake. Minus 9. So minus 9 plus 9 will be cancelled. So multiply this, you will get 2.244 meters. 2.244 meters you are getting. So ultimately, so what is the approximate? 2.244 or it is 2.2 meter. Original calculation you are getting 2.244 meter. In the NCERT, in this molecular basis of inheritance, under the heading of packaging of DNA helix, they gave that approximately 2.2 meter. But in the cell chapter, under the heading of nucleus, they gave approximately 2 meter. They gave all these things are same only. Originally, you are getting 2.244. You can approximate to 2.2 or you can approximate to 2 meter. But what is the length of this? Is it the length of single chromosome? No. In single human diploid cell, total 46 chromosomes are there. If all the 46 are attached back to back, then this length will be 2.2 meters. So this 2.2 meter or 2 meter is the length of DNA that is present in single human diploid cell. That means for all the 46 combinedly it is 2.2 meters. This is the information is. Very good. Now you need to link up the information. What is the information? The length of DNA present in one cell is 2.2 meter. But this DNA is present within a small nucleus and its diameter is approximately 1 micrometer. 1 micrometer means 10 to the power minus 6 meter. But this one is 2.244 meter. And in the cell chapter we studied what is the average size for eukaryotic cell? 10 to 20 micrometer. Now understand this. Size of cell is small and inside that nucleus is very small. But inside that the DNA length is very long. This DNA is very long, yes or no? Then the question here is how this much of long DNA is present in very small nucleus. This is the question. How this much of long DNA is present in very small nucleus. That's why it should undergo some packaging. It should be packaged, kept very condensed. Then only it can be kept inside the nucleus. So how exactly it is undergoing the packaging? So that we need to study. That's why they are talking about the DNA packaging. Understood or not? This is for eukaryote. Now we will talk about the prokaryote. But before this, there is one more question. Now understand this. For the human diploid cell, this is the value. Suppose they are asking about the human haploid cell. So diploid cell means they can directly ask diploid cell, or they may ask that it is somatic cell, or they may ask gamete mother cell. Gamete mother cell is diploid cell. This is the value. Suppose they are asking about the gamete directly, sperm cell or ovum directly they are asking. Then what you will write? It will be 3.3 into 10 to the power 9 base pair. So exactly half of this. So for the haploid, what will be the answer? It is 1.1 meter, or if you want exact 1.122 meter. Remember that value also. Okay. Now come to the next one. Now we will calculate with respect to this is human means eukaryote you are talking. E. coli means it is prokaryote, bacterium you are talking. So both the things we need to study. That's why. So what is the length of E. coli DNA? So in that table wherever we studied that circular double strand DNA is there they have given this. What is the length they give? 4.6 into 10 to the power 6 base pairs they give, and multiply with 0.34 nanometer. So it is multiply with nano means 10 to the power minus 9. Okay, here I made correct, but here there is a small mistake. So please change it. Okay, I am sorry. So now you just simplify this. What is the value you are getting? 1.564 millimeter. It is not meter, millimeter. Okay, for human you are writing in meters. Better try to remember these values. Sometimes they may ask same as it is question in examination. So at that time, don't waste time in calculating all these things. At least remember these values. Okay. For this E. coli, for 4.6 into 10 to the power 6 base pairs, how much we are getting? 1.564 millimeter we are getting. Understood or not? So for 4.6 into 10 to the power base pairs, it is 1.564 millimeter. Here also same question. In the cell chapter, we studied what is the average size for the bacterium. So in one paragraph, they gave 3 to 5 micrometer. In the diagram, they gave 1 to 2. So we have made it as average of 1 to 5. 1 to 5 micrometer is the size of bacterium. But the DNA present inside the bacterium is 1.564 millimeter, means it is more than 1000 times longer. The DNA bacterium is smaller and the DNA present inside this is 1000 times longer than the length of this. Then the question is how this much of long DNA is present inside the small bacterium. Same is the case with eukaryote also. So now in whether prokaryote, eukaryote, whatever it is, the DNA presence inside that is much longer when compared with the cell. Now the concept is how this much of long DNA is present inside the small cell. For that we need to study the packaging. Understood? But before directly going with the packaging, now they have given one question. Please understand this. This question was not given in NCERT. What is the NCERT question? This is the question. If the length of E. coli DNA is 1.36 millimeters, then calculate the base pairs. Reverse they are asking. Many students, without calculation, they are writing that it is 4.6 into 10 to the power 9 base, 10 to the power 6 base pairs. Why they are writing like this? They are writing that 4.6 into 10 to the power 6 base pairs. Without calculation, they are doing that is wrong. Please observe carefully. If it is 4.6 into 10 to the power 6 base pairs, what is the length you got? 1.564 millimeter. But here they asked 1.36. For 1.564, if it is 4.6, for 1.36 will you get 4.6? No, definitely it is not. Definitely it is lesser than this. For 1.56, 4.6 means for 1.36 definitely lesser than this. So you need to calculate this. Please do the calculation. This is direct NCERT in-text question. So if they are giving in millimeter, asking the base pair, then what you need to do? Now you need to go for the division with 0.34 millimeter. If base pairs are given and they are asking in the other units, then you need to multiply with 0.34. If it is the reverse, divide with 0.34. So 1.36 millimeter divided by 0.34 nanometer. So simplify this. So you will get 4 into 10 to the power 6 base pair. It is not 4.6 into 10 to the power 6. It is simply 4 into 10 to the power 6. Sometimes they may give 4.0 into 10 to the power 6. That is also correct. 4.0 and 4, both of them are same only. So 4 into 10 to the power 6 is the answer. So these are some standard questions. You need to know the answer. In the examination, if they are using as it is value, suppose, you should not waste time in calculating there. You should go for the answer. So that's why what you do is number of base pairs and the size. For human diploid, human haploid, in the equally also, if it is 4.6 into 10 to the power 6, what is the value? If it is 4 into 10 to the power 6, what is the value? Make a table in the NCERT textbook itself. So every time you just revise this so that it may be useful. Clear? So this is what you need to do it. Nice. So now this is the basic calculation. From this calculation, what is the question that is arising? Size of the cell is smaller and the DNA inside that is larger. Then how this much of long DNA is present inside a small cell? For that we are studying about DNA packaging. First we are talking about the prokaryotes. In the case of prokaryotes, it is a very simple type. Here histone proteins are not present. DNA is having some coils, also called as loops, and these loops are stabilized by some proteins. Technically these proteins are called as nucleoid associated proteins because the DNA itself is called as nucleoid, and here the location where this DNA is present is also called as nucleoid. In the NCERT very clearly they said a point. In the case of prokaryotes, nuclear membrane is not there. DNA is present directly in the cytoplasm. Even though it is directly present in cytoplasm, it is not spread in the entire cytoplasm. It is located in a particular location. Location is called a nucleoid. Even the location is called nucleoid. Even the DNA is also called as nucleoid. And to that nucleoid, some basic proteins, positively charged proteins are associated. The reason here is why positively charged proteins. Why positively charged proteins are required? You need to understand the logic here. This logic is same for both prokaryote and eukaryote. Whether prokaryote, eukaryote, wherever it is, this DNA is negatively charged. DNA is negatively charged. Why DNA is negatively charged? DNA is having nucleotides. In the nucleotide, nitrogenous base, sugar and phosphate, phosphoric acid, acidic nature is there. H3PO4. In that H plus will be removed. So H2PO4 minus. So DNA is getting minus, negative charge. DNA is a negatively charged molecule. Whether it is a small molecule, large molecule, whatever it is, DNA is always negatively charged. So when this negatively charged DNA, when it is trying to pack together, what happens? Negative repulsions will occur. So it cannot come very closely. So to neutralize the negative charge, you need to add some positive charge. Positive means basic. That's why we need basic proteins. There are different types of basic proteins in the nature. And here in the case of prokaryotes, they are called as nucleoid associated proteins, which are positively charged proteins. Simply DNA is making some loops, and between these loops some basic proteins will come so that they will neutralize the charge, so that DNA can be kept very closely. So in this way it is packed, and now it is present inside the cytoplasm. That's it. So very simple type of packaging we are observing in prokaryotes. But in the case of eukaryotes, it is highly complicated. In the case of proteins, there are some specialized positively charged proteins called as histones are present, and these histones are getting positive charge because of more and more lysine and arginines. Lysine and arginine are basic amino acids, positively charged amino acids. So histones are rich in basic or positively charged amino acids called lysine and arginine, and these positively charged proteins are interacted with eukaryotic DNA. So eukaryotic DNA is organized into chromosomes, and this point once again I am linking with cell chapter. There also I gave very clear cut explanation. The term chromosome is useful only for eukaryotes. It is not used for prokaryotes. Very clearly I am telling you this. The term chromosome is used only for eukaryotes, not for prokaryotes, because in the case of eukaryotes only histone proteins are interacted with DNA, and here only we are observing the chromosomes. The term chromosome is not technically applicable to prokaryote, but in the cell chapter within bracket they give prochromosome term. Explanation I gave very clearly in that particular part. So you just go and revise that particular thing. Now come back to this one. In the case of eukaryotes, how DNA is associated with histone proteins? In the case of eukaryotes, total 5 types of histones are there. H1, H2A, H2B, H3, H4. At least H1 name, they have at least given in the diagram of NCERT. So please remember this. And remaining 4, what are these 4? H2A, H2B, H3 and H4. And actually each one is 1 unit. So 1, 2, 3, 4. And like this, another 4 are there. So that means each one is present in 2 copies. H2A 2 copies, H2B 2 copies, H3 2 copies, H4 2 copies. So total 8 copies we are getting. All the 8 are present together. It is called as histone octamer. Sorry. It is called as histone octamer. Now around this octamer, now DNA will be coiling and it is making approximately 1 and 3/4 coil. That means one complete coil and the other coil is not complete. It is having approximately 3/4 coiling. So in this way 1 and 3/4 times it is coiling around histone proteins. Since negatively charged DNA is interacting with positively charged histone octamer, charge will be neutralized. So DNA can be compactly packed. So DNA compaction or DNA packaging can be easily done. So this is how they are present here. And now here there is a length of DNA that is connecting two adjacent octamers is called as linker DNA. And generally, so in one histone, that means from here to here and up to this. Starting from here, coiling around this and coming up to this. As per NCERT, from here to here it is called as one nucleosome. In the one nucleosome, the length of DNA present is, in one nucleosome, the length of DNA present is approximately 200 base pairs. Out of these 200 base pairs, length of linker is approximately 54 base pairs. So, the length of DNA that is contacted or connected directly with histone octamer is approximately 146 base pairs. So this is how the arrangement is present, and this particular structure is called as a nucleosome. DNA that is coiled around the histone octamers is called as nucleosome, and nucleosomes are the repeating units. Likewise, several millions of these nucleosomes are present. Clear? And these nucleosomes we can clearly see them as beads on string structure under electron microscope image. So in the electron microscope structure we can see them as beads on string structure. These beads are nothing but the nucleosomes, and the string that is connecting all these is only the DNA. So it is the DNA that is connecting all these nucleosomes. Is it clear? And even around this histone octamer also this DNA is present. That's why this is entirely called as nucleosome, and the linker is nothing but DNA. So in this way, the bead-like structures are nothing but nucleosomes, whereas the string means the thread-like structure that is connecting is the DNA. So this is what that they are saying here. So observe carefully, and the interphase chromatin will clearly see this beads on string structure. Clear? So that's why in the interphase chromatin, so these nucleosomes are the repeating structures. Clear? Next one. Out of 200 base pairs, in one nucleosome 200 base pairs length is present, 146 base pairs is approximately interacted with histone octamer, and remaining 54 base pairs is called as the linker DNA. Clear? And in the NCERT they have asked a question, theoretically how many such beads means nucleosomes are present. How will you do the calculation? For the human diploid cell, take the number of base pairs, 6.6 into 10 to the power 9 base pairs. In one nucleosome, how many base pairs? 200 base pairs. So divide with that, and you will get number of nucleosomes. How much? 3.3 into 10 to the power 7 nucleosomes. So simplify this. You can write this as 33 into 10 to the power 6 nucleosomes. 10 to the power 6 means what? Million. So you can write that 33 million nucleosomes are seen in human diploid. If the same question they are asking for human haploid, then it will be half of this, 17.5, not 17.5, 16.5. 16.5 million nucleosomes will be the answer. So in this way also questions can be asked. So please observe them very clearly. Okay. Next one, the number of nucleosomes that we are observing in E. coli. Suppose for with respect to human they asked, you did the calculation, but if they are asking with respect to the E. coli, then will you take that 4.6 into 10 to the power 6 divided by 200? No, you cannot take it, because the nucleosome concept is not applicable to prokaryote. That point also you need to observe. E. coli is a prokaryote. So in the case of prokaryote, nucleosome concept is not there. For nucleosome concept, histones are required, but histones are not present in prokaryote. So that's why nucleosomes are not present. So simply whatever is the organism, prokaryote they have given, or sometimes they may give the virus. So whatever they are giving, so in those organisms we are not observing nucleosomes. Nucleosomes are seen only in the eukaryotes. So please observe this. That's why simply you will write that in the E. coli number of nucleosomes are 0. That's it. Clear? Next one. This is what, if it is only DNA, If it is interact with histone proteins, this is how it is forming nucleosomes. They will again undergo coiling, and that will undergo further coiling, that will undergo further coiling, and finally it is forming the metaphase chromosome. So maximum condensation we are observing in metaphase chromosome. Yes or no? For the structure study, the metaphase is the best phase. Yes or no? In the cell cycle, cell division chapter, we have very clearly studied. So this is the only DNA, histone proteins. Interaction will from the nucleosomes, and then they will undergo coiling and form solenoid form, anyhow not there in NCERT, and that will undergo further coiling and form supersolenoid, and that will undergo further coiling, and finally it is forming the metaphase chromosome. Okay, so actually the supersolenoid form is the normal chromatin phase. That's it. In that form it is actually present. Nice. Next one, regarding the composition of the chromatin, if you see in the NCERT, in the cell chapter under the nucleus they have mentioned this. In the chromatin, if you see, DNA is present, histone proteins are there. These are the major things. Along with this, even RNA is also there, even HD proteins are also there, and even some minerals are also present, even some trace amounts of lipids are also present. Many of these are present, but DNA only is the genetic material. Please understand this. Clear? And regarding the non-histone chromosomal proteins, anyhow, along with this DNA, histone proteins are present. Not only histone proteins, some other proteins, they are not histones. There are some other proteins, they are also present along with this. They are called as non-histone chromosomal proteins. Next important point we are studying is euchromatin and heterochromatin. So euchromatin are loosely coiled portions, heterochromatin are tightly coiled portions of the chromatin, and very clearly in the NCERT they have given some information. So euchromatin are loosely packed regions of chromatin, and they are stained lightly when we are using the dyes. Okay, and then they are transcriptionally active, means transcription can occur here because they are loosely coiled. RNA polymerase can easily come and attach, and it can do the transcription. And here the replication of this particular euchromatin will occur in early S phase. And if we are talking for the heterochromatin, it is densely packed, means tightly, very tightly packed. So that's why here what happens, the density means the more DNA is present in the less space. So more DNA is present means it will take more stain. That means it will stain darkly. And this is transcriptionally inactive. Why it is transcriptionally inactive? Since it is very tightly coiled, RNA polymerase cannot go there. So that's why it is transcriptionally inactive. Transcription cannot occur there. And these portions will divide during late S phase, means replicate during late S phase. This is what some basic information that they have given for the packaging of DNA helix. And then they are talking about the properties of the genetic material, and here they are saying that in the majority of the organisms, DNA is the genetic material. DNA is the genetic material in all 5 kingdoms, and some viruses like Bacteriophage lambda, Bacteriophage 5x174, and T2 Bacteriophage. In these NCERT examples, we are studying the DNA is the genetic material. But later on, scientists have proved that some viruses are having RNA as the genetic material. Standard examples are tobacco mosaic virus and then QB bacteriophage. These are some standard examples. Not only this, along with this, you can add novel coronavirus 2, and which is causing COVID-19. This is also RNA virus, RNA containing virus. Very important. So please remember this. So in this way, even some viruses are having RNA as genetic material. That's it. But whether it is DNA or RNA, only these two. Not the proteins, lipids, carbohydrates, they cannot act as genetic material. So in this way, the molecule that is acting as genetic material should follow some properties. So what are these properties? 4 important requirements they gave in NCERT. First one, it should be able to generate its replica. That means it should undergo replication. So who can undergo replication? DNA can undergo replication, RNA can undergo replication. Can proteins undergo replication? No. Can carbohydrates undergo replication? No. Can lipids undergo replication? No. So in the first criteria itself, they are failing. So they are not useful. These 3 are not useful for genetic material. Only DNA and RNA are useful for genetic material. First thing. Second important thing, it should be stable chemically and structurally. So who is more stable? So anyhow, forget about the remaining 3. Now our comparison is between DNA and RNA only. So between DNA and RNA, who is more chemically stable, chemically and structurally? DNA is more stable when compared with the RNA. So reasons also they gave. So the molecule that is acting as genetic material, it should be stable in the lifetime of organism, whether it is in the embryonic stage or early childhood or in the adulthood or in the old age, whatever it is, whatever is the life cycle. In some insects, you will find that there are some stages like larva stage, pupa stage, adult stage, likewise they are there. So whatever is the stage, in all these stages, the genetic material should be stable. And for this particular property, both DNA and RNA will follow. Next important point is temperature stability. If you are talking with respect to Griffith experiment, in the Griffith experiment, Griffith killed the bacterium by exposing to heat. At that time what happened? The double stranded DNA the two strands got separated. But upon cooling, again it got reattached. Bacterium is dead, but the DNA is again getting double stranded DNA, so it is still active. That's why the DNA from heat killed S is able to go to S strain, and there, it can go into R strain. There it can transform R into S strain. That means even after exposure to heat also the DNA is still working. That means DNA is resistant to heat exposure. But RNA is not like that. RNA is labile. Labile means it is unstable. Even a small exposure to heat also will completely damage the RNA. This is what the problem is. Understood? Next one, with respect to the structure, if you are talking, so DNA in the structure, it is having deoxyribose. Because of the deoxyribose, it is more stable. But in the RNA, it is having hydroxyl group in the second carbon of ribose sugar. So hydroxyl group is highly reactive group. It is involving in so many reactions. Not only that, RNA as an enzyme, it is involving in so many reactions. Ribosome examples we studied. So in this way, RNA is less stable. Understood or not? Next important point is, with respect to DNA, in the case of DNA, thymine is present. So presence of thymine is giving some extra stability in the repair mechanisms. But in the case of RNA, thymine is not there. So for RNA, repair mechanisms are very few and they are not that much efficient. So that is the reason why DNA is structurally and chemically more stable when compared with RNA. So these are the various reasons that they have very clearly mentioned in NCERT. Clear? Next one, it should provide the scope for slow changes. Remember, requirement itself is the slow changes are required. But RNA will give rapid or fast rate changes. That's why slow changes are not given by RNA. They are given by only DNA. Please remember why slow changes are required. What is the problem with the rapid or fast changes that are given by RNA? If you see RNA containing viruses, let us take the example of COVID-19 only. So that in the case of COVID-19, that novel coronavirus, before it is stabilizing one particular strain, it is converted into another strain. That means stability is not there. If stability is not there, how it is useful for evolution? Before see, in the evolution what should happen? First one species should be stabilized. Slowly, along with this, another should be developed. In this way, many species should be developed one after the other. That is actually required for evolution. But in the case of RNA, because of the fast and rapid mutations, that stabilization is not possible. So that's why RNA containing viruses will evolve faster. They are evolving much quickly, but they are not useful for stabilization. So that's why they are not useful for evolution process. So that is the reason that they are saying here. So that's why for the evolution, slow changes are required. And since DNA is having repair mechanism, even though mutations are occurring, because of the repair mechanism, I am not saying all mutations, majority of the mutations will come back. So in this way, very few or rare mutations will be left over. So in the course of time, slowly, these changes will accumulate, and finally that may be useful for evolution of a new species along with existing species. The original species will be existing, and from this slowly after millions of years, a new species will be developing. Understood or not? In this way, DNA genetic material, DNA, if it is acting as genetic material, that is much more useful for evolution when compared with RNA. Next one, it should be able to express itself in the form of Mendelian characters. What is the meaning of this? In the Mendelian characters means what? It should have some genes, and the genes should undergo expression so that they should control some characters, and they should move from one generation to next generation. And this is done by both DNA and RNA. So in this way, overall, if you see, first and last are satisfied by both DNA and RNA. Second one, if you are observing stability, this is the most important factor. So DNA is much more stable when compared with RNA. And anyhow, for the evolution, slow changes are required. They are supported by DNA, not by RNA. So overall, if you see, even though both DNA and RNA can act as genetic material, again between these two, DNA is the better. So that's why for the storage of genetic information, DNA is the best. But for the transmission of genetic information, RNA is a better molecule. So in this way, in the evolution, what happened? So that's why if you see, only in some viruses RNA is genetic material. Remaining viruses, DNA genetic material. All 5 kingdoms, DNA genetic material. So for the genetic information storage, DNA is important. That's why DNA is the genetic material. And from the DNA, if proteins are directly produced, there are some chances of, if controlling points are more, the regulation will be more. So that's why in the evolution what happened? So from the DNA, RNA is coming, from the RNA proteins are produced. That's why for information storage DNA is the best, and for the transmission of information RNA is a better molecule. So with this particular point is also completed. Then they are talking about the RNA world concept. What is this RNA world concept? Initially in the beginning of the evolution, it is not the DNA, it is the RNA that was evolved first. Please understand this. Even though RNA is less stable, RNA was evolved first. RNA was the first genetic material. First genetic material was RNA. But later on what happened? So since RNA is not that much stable, and since it is not allowing, it is not that much useful for evolution, it is not allowing the evolution, means it is allowing for quick evolution, but the original previous species is not getting stabilized. That's why what happened is, in the RNA small chemical changes occurred. What type of changes? Ribose sugar converted into deoxyribose, uracil is converted into thymine. In this way, because of slight changes, RNA got converted into DNA. So once DNA got produced, now it has stabilized this. So in this way, now that remaining all organisms got developed with help of DNA molecule, and they contain DNA as the genetic material. But in the beginning, RNA is the major molecule. So RNA acted as genetic material. Not only genetic material, RNA involved in so many processes. RNA acted as enzyme also. So that is the reason why initially in the course of beginning of the evolution, RNA is the major molecule around which majority of these processes were developed. But later on what happened is, slowly its function was reduced and other molecules got developed. Initially RNA was the genetic material, later on DNA became genetic material. Initially RNA was the enzyme, but later on proteins were developed from that. Yes or no? Proteins were developed, and proteins took over the function of enzymes. So that's why nowadays if you are observing, there are very few examples where we can find RNA is genetic material. There are very few reactions where we can find RNA is the enzyme, because slowly they have got changed. But in the beginning of the evolution, RNA was the major molecule. Around the RNA majority processes were developed, and all these concepts will support the RNA world concept. That's it. So that's why today also, wherever you are studying the example of RNA genetic material, wherever you are studying that RNA is acting as an enzyme, all these things will support the concept of RNA world. Please remember this particular sentence, because this sentence once again I will link up with a transcription in the second part of this MBA revision. Clear? So please remember this. Next we are studying about the central dogma of molecular biology. Who proposed this? It was originally proposed by the Crick. Crick proposed this central dogma in molecular biology. What actually he proposed? He proposed that from the DNA, RNA is produced, from the RNA, protein is produced. That's why the information flow is always unidirectional. Not only this, from the DNA again, DNA can also be produced. This is what the information that they have, that Crick has proposed. Okay. But even though in the majority of organisms this is happening, there are very rare cases where the reverse is happening. What is that reverse? From the RNA, DNA is produced, and this process is opposite to transcription. Since it is opposite to transcription, it is called as a reverse transcription. The enzyme that is required for this process is called as reverse transcriptase, and the viruses in which this process is taking place are called as retroviruses. Retro means reverse. Since reverse transcription is occurring here, so they are called as retroviruses. One standard and very important example for retroviruses is HIV, human immunodeficiency virus. Regarding this, in the human health and diseases, very clearly they mentioned, so please remember this. Clear? Very good. Next one, and it was originally discovered by Temin and Baltimore individually. So reverse transcription is also called as a Teminism. Teminism, if they are giving, Teminism is nothing but reverse transcription. Clear? Next one. Next we are talking about the DNA replication process. Who first proposed it? Sentences are very clear. Who first proposed DNA replication? Who first proved DNA replication? All these are very important. Who first proposed DNA replication? It is Watson and Crick. Who first proved this? It is the Meselson-Stahl. Who first proved in prokaryote? It is Meselson-Stahl. In what is that prokaryote? E. coli bacterium. Next one. Who first proved in eukaryote? It is Taylor and co-workers. And what is that eukaryotic organism? It is the Vicia faba, a plant. Understood or not? And who first proved in chromosomes? Please remember this one. This is a technical word, technical question. Who first proved in chromosomes? Chromosomes term is applicable only for eukaryote. So who first proved in eukaryote? It is the Taylor. So who first proved DNA replication in eukaryotes that it is semi-conservative? It is the Taylor and co-workers. So in this way different questions can be asked. So first I am using a word called proposed. Who first proposed about the DNA replication? It is the Watson and Crick. And what they proposed? They proposed that since there is a complementary pairing here, the two parental strands will be separated and each one will act as a template for synthesis of new DNA molecule, so that the two newly produced molecules will be identical to the parent DNA molecule. And this is nothing but semi-conservative DNA replication. This is what they proposed. And simply they proposed based on the complementary pairing, but they did not give any proof. So that's why who first proposed semi-conservative DNA replication is Watson and Crick. With that one is over. But now we are talking about who first proved. This is very important. Who first proved that DNA replication is semi-conservative? It is the Meselson-Stahl. Please observe this. And what is the organism? It is the Escherichia coli, E. coli bacteria. And in the medium, they have used N15. Please remember, N15 is heavy isotope. It is not radioactive isotope. N15 is heavy isotope. It is not radioactive isotope. Please remember. So in this way, N15 containing ammonium chloride, they have used in their experiment. So what is this particular experiment? So that we need to understand. This is the NCERT diagram. Now we will understand this very clearly. Observe this information carefully. So in the regular classes, I have given the full explanation. Here in simple, I will try to explain this. So initially they have taken the E. coli in N15 containing medium. Okay. So initially they have grown this in N15 containing medium. So here they have grown the bacterium for several generations. If they are growing this in N15 containing medium for several generations, what happens? Each and every component that is having nitrogen in E. coli will get that N15. So in this way, please remember and read the NCERT sentence carefully. Not only DNA of E. coli, even carbohydrates, proteins and other compounds, wherever nitrogen is present, they are also getting the N15. But we are not worried about other compounds, because ultimately after the growth of bacterium, from the bacterium we are isolating double-stranded DNA only. Since we are isolating double-stranded DNA only, even the N15 is going into other compounds, we are not worried. So there is no problem with our experiment. So even though that is going into that, our main focus will be DNA only. So since you are growing the bacterium in N15 medium for several generations, both the DNA strands will get N15. And here what we are doing, we are isolating DNA from this. We are subjecting to density gradient centrifugation. Clear? So this is what we are doing. This is the normal bacterial leaflet. This is the N15 medium, heavy medium. So here you are getting only single band, and this single band is corresponding to heavy DNA because it is having both N15 and N15. Forget about this particular one. Start from this. So in this way, initially they have grown in N15 medium. Then what they did? Then they have transferred bacteria to N14 medium. From here onwards, every time it is N14 medium only. Please remember this. And here, after every 20 minutes, they have collected the sample. After transfer, 20 minutes, another 20 means 20 plus 20, 40. After 20 means 40 plus 20 will be 60. After 20 means 60 plus 20, 80 minutes. In this way, for every 20 minutes, they have started collecting the bacterial cells, some bacterial cells. Why they are doing like this? Because E. coli bacteria will divide for every 20 minutes. Because of this, every 20 minutes, what happens? The number will be doubled. So here I made a table also. If one bacterium is there, with respect to one bacterium, I am calculating. One bacteria will become 2 here, 2 will become 4 here, 4 will become 8 here, 8 will become 16 here. In this way, number will be doubled. Yes or no? So accordingly here, we can understand the number of DNA molecules and the bands also. So what they did is they have transferred E. coli to N14 containing medium. In that medium, after 20 minutes, they have collected some cells. From that, they isolated DNA and subjected to cesium chloride density gradient. This is also very important. Cesium chloride density gradient medium. So here also they got only one band. But please understand, the level for this band and this band are not same. This is actually a little bit higher. That means density of this one is lesser than this. And technically actually it is having hybrid. That means one strand is N15, other strand is N14. Clear? And after another 20 minutes, isolated DNA and subjected to density gradient. And one band is exactly matching with this, but another band is little bit above. And here they got two bands, and one is corresponding to hybrid, and other one little bit above is the light DNA having both N14. And after another 20 minutes, same two bands. But observe carefully, when compared with the thickness of this band thickness is 3 times more. 3 times more. Because now the number of light DNA molecules are keep on increasing. After another 20 also 2 bands are there, but here it is 1:7 ratio. So in this way what happens, the thickness of this DNA band, and if you are talking with respect to this one, it is almost constant, but this one is keep on increasing, light DNA percentage is keep on increasing. And why this is happening, how exactly it is changing, and this particular result is matching with semi-conservative DNA replication. So in this way they proved that DNA replication is semi-conservative, but here in the revision I cannot spend more time than this, so I hope you can understand this. But in the regular class I told you what is conservative, what is semi-conservative, what is dispersive, how this result can be compared with conservative, semi-conservative, dispersive, then how finally you will prove that this is semi-conservative. Everything was explained very clearly in the regular classes, so here we cannot spend much time more than this. I hope you can understand this. In this way they perform the experiment. Based on this experiment they conclude that this result is matching with semi-conservative DNA replication. So in this way they proved that DNA replication in E. coli, that is a prokaryote, is semi-conservative DNA replication. That's what they proved. Clear? And here what you need to observe is, when it was initially grown in N15 medium, only one band, that is heavy DNA. After transfer to N14 in the first generation, after 20 minutes means first generation, here also only one band. What is this one? Only hybrid. So hybrid DNA after transfer to N14, every time hybrid is there, but heavy is never present. Once it is transferred to N14, heavy is never present. Please remember, all these are important conclusions. Heavy is never present, hybrid is always present. And light, presence of light is started in second generation. From then onwards, every time it is also present. And next one, this thickness of this heavy hybrid DNA, thickness of this hybrid always remains constant, but the thickness of this light is keep on increasing. But if the same thing, if you are writing in the percentage here 100% hybrid, here 50% hybrid, 50% light, 25% hybrid, 75% light. So percentage wise if you see, for the hybrid percentage is decreasing, for the light percentage is keep on increasing. So in this way, same information, same diagram, question can be asked in different ways. So read the question very carefully, so accordingly you will get the answer. Next one here, what is the ratio between hybrid and light? It is 1:1. Hybrid and light 1:3, 1:7. So in this way they can ask. So here the number of molecules will decide the thickness of the band. The same question with respect to number of molecules, here they can ask what is the ratio between thickness of the bands, 1:1, 1:3, 1:7. It all will be decided by number of molecules. That's it. If you know the number of molecules, accordingly you can do it in the regular class. So since we get the more time, so there I have given the clear explanation, and here we are going with some summarized conclusions. That's it. So this is what the experiment, and for this experiment the same thing you can write in different representations. Everything, all these are related to the same thing, only in different representations you can write all these things. Clear? So this is what, and here I made a table also, ratio between heavy, hybrid, light. So when it is in N15 medium, then only you can see heavy, and here hybrid and light are there, not there. Once transferred to N14 medium, first generation, second generation, 20 minutes, 40 minutes, and then 60, 80, 100, 120, likewise different minutes I have given. And if you are initially starting with one bacterium, after 20 minutes here it will, 1 will become 2, 2 will become 4, and then 8, 16, likewise number is going up. And here heavy is present only when it is in N15 medium. Once transferred to N14, heavy is never present. Heavy is never present. And here according to this number, so how many hybrid, how many light I have given. And once here I have taken only hybrid and light, only last two. You just simplify this. 2:2 means 1:1. Here 2:6 means 1:3. 2:14 means 1:7. Likewise I have given the information with respect to hybrid and light here. Within brackets I give percentage wise also. Questions can be asked in any ways. So if you are able to draw that particular diagram on your own, then you can write all these things very simply. Clear? So please practice all these things. Not only this, the questions can be, application questions can be asked in reverse. In the original experiment, initially grown in 15, then transferred to 14. So they may ask in reverse. Understood or not? And original experiment is with respect to semi-conservative. They may ask that if DNA replication occurs in conservative manner, then what is the answer? So in this way, all these things actually we have discussed very clearly in the regular lecture. So please go through that. And here we are going with only NCERT based information. So this much is enough. And then there is an in-text question. If E. coli were allowed to divide for 80 minutes, then what will be the, observe this, proportion of, in the sequence wise they asked light first and then hybrid then. After 80 minutes, you need to write this particular one. After 80 minutes means you just see here. This is what after 80 minutes. So how many number of molecules you are getting? 2 molecules of hybrid you are getting and 14 molecules of light you are getting. Understood or not? And here what they have asked, hybrid to light 14. So sequence wise you see, light they have asked, so 14 is to hybrid 2. So 14 is to 2 means 7 is to 1. This is how we need to do. Here I gave the formula also, how to use this formula, explanation, everything I gave in the regular lecture. So please go through this. Okay. Next one. So with that, the DNA replication is semi-conservative in E. coli or in prokaryote was proved by Meselson-Stahl. That experiment was completed. Now here we are talking about some scientists called as Taylor, Woods and Hughes. So their experiment, it is simply called as a Taylor et al. Taylor et al means Taylor and co-workers technically. So Taylor and co-workers and their experiment. So these scientists have performed experiment on Vicia faba. It is also called as broad bean or faba bean or faba bean, whatever it is. It is a plant. So in this plant they performed this. Since it is a plant, it is a eukaryote. Sorry. It is eukaryote. So in the eukaryote, chromosomes are present. So who first proved that semi-conservative DNA replication? DNA replication occurs semi-conservatively in eukaryote. Eukaryote, it is Taylor and co-workers. In the chromosomes, Taylor and co-workers. This is what the information is. But in the previous experiment and here there is a small difference. In Meselson-Stahl experiment, they used N15 non-radioactive isotope. But in this experiment, they used tritiated thymidine, which is a radioactive isotope. Since it is radioactive isotope, here density gradient centrifugation will not work here. So here you need to go for some measuring the radioactivity. For that there are different counters are there. So those we are using, and you don't need that particular one. They will measure the radioactivity. They will measure the radioactivity in such a way that whether radioactivity is present or not. That also they will measure. If present, how much it is present. Quantification also they will do it. This is what the experiment is. And here in NCERT they have not given much information. Let us imagine that this is what the DNA molecule is. So in the chromosomes what happens? So in the next every time, here also semi-conservative replication is there. So in every generation what happens? Initially they have supplied tritiated thymidine for several generations to the plant root cells. So that now these cells are continuously taking. So both the DNA strands will get radioactive DNA, radioactive isotope. And after some time they have stopped the supply of radioactive thymidine and they have given the non-radioactive. Radioactive is called hot thymidine. Non-radioactive thymidine is called cold thymidine. They have started giving the cold thymidine. Then what happened? So now they have observed this result, and this result is exactly matching with whatever they have observed in the Meselson-Stahl experiment. For this one also very clear-cut explanation I have given in the regular class, because here we need to draw so many diagrams for the clear understanding. So in the regular class I have given this. In conclusion, I am telling you, so this result is also exactly matching with whatever the result they have observed in the case of Meselson-Stahl experiment. So here also I made the table, but here the radioactivity, number of cells with radioactivity and non-radioactivity, and these ratios also exactly they will match with the table whatever we made for Meselson-Stahl experiment. So in this way, these scientists also proved that DNA replication in eukaryotes is also semi-conservative. That means DNA replication in chromosomes also semi-conservative. It was proved by Taylor and co-workers. Clear? And now for this particular video, this is the last part. Now we are going with the DNA replication. In the NCERT, please observe the heading carefully. They are talking about the enzymes and machinery. That's it. Enzymes and machinery, only that much is our syllabus. You don't need to worry about how exactly DNA replication is initiated, what is the exact process and everything. You don't need to understand. Simply understand what is the exact information given in NCERT, that much is enough. That's it, over. So that's why I am also closing like that only. But in the regular class, what I did is, along with this particular information, I gave the sequential flowchart procedure. I have explained also. I have provided you notes also. Everything was there in that regular classes, because if you understand the sequence of the process, then you can interlink all these informations at a stretch. So that's why in the regular class, since we have more time, I did that. But here, since it is only the machinery, I will simply explain each and every point related to NCERT, but I may give 1 or 2 extra points also for better understanding, and there we will close this particular one. So in the DNA replication, different components, mainly different enzymes are there, many other proteins are involved, and even nucleotides, many things are required here. So first we will go with the basic information. If you are talking about DNA replication in prokaryotes, the important point we need to interlink with cell chapter. There also I told you. So here we are interlinking once again. In the cell chapter, for the prokaryotes, when we studied about mesosomes, there they have given the functions of mesosomes. Mesosomes involve in DNA replication and distribution to daughter cell and cell wall formation. This is what they give. So now we will talk about the mesosome. Inward folding, infolding of plasma membrane is the mesosome. Generally, this nucleoid is attached to the plasma membrane at the mesosome. And now what happens? This DNA will undergo replication. After DNA replication, each one copy will attach on one side of mesosome, other copy will attach on other side of mesosome. Then mesosome will undergo folding. And now this invagination will extend and extend, it will touch the other side so that it will divide into two bacteria. So in this way, two bacteria are formed. And now what happens? Wherever it got separated, there the cell wall also will be formed. So in this way, this mesosome helps in DNA replication and distribution to daughter cells, and also at that location cell wall will be formed. In this way, two daughter bacteria are produced, and again they are undergoing this particular bacterial cell cycle. Understood or not? And please remember, in the cell cycle chapter, I have very clearly explained you G1 phase, S phase, G2 phase, M phase, these are not applicable to prokaryotes. These phases are applicable only for eukaryotes. So in the case of this bacteria, what we are studying, only two things: DNA replication followed by bacterial fission. DNA replication followed by bacterial fission. Only two things are there. This is what happening every time. Clear? So in this way, bacterial DNA replication is taking place. But in the case of eukaryotes, DNA replication occurs in a specific stage. That stage is called as S phase. Only in the S phase DNA replication will occur. Is it clear? But please remember, in our syllabus of NCERT class 12, we are studying DNA replication with respect to prokaryote only. Please remember this. Very clearly I am telling you. We are studying information with respect to prokaryotic DNA replication only. So please observe this. Now we are talking about the origin of replication. In the case of bacterial DNA, there is a specific location, and that location is called as origin of replication, because this is the place from where replication will be originated. Every time, whenever DNA replication occurs, it should begin from this one only. And this is a fixed location. Every time it will start from there only. That's why it is called as origin of replication. That's why origin of replication is very, very important in biotechnology. So that's why in the NCERT also they have given a sentence. So if you want to propagate a piece of DNA, then vector has to provide this origin of replication, because in the case in the biotechnology, our gene of interest is not attached along with this. Means whenever we are transferring gene of interest, only gene of interest you are transferring. Along with gene of interest, you are not transferring origin of replication, because origin of replication will be provided by vector itself. So vector has to provide origin of replication, so that a piece of DNA, means alien DNA, will be transferred and it will be attached to origin of replication somewhere in the downstream of this origin of replication we are attaching, so that it will be undergoing the replication. That's it. So in this way you can make multiple copies. You can propagate that piece of DNA using recombinant DNA procedures. This is what they are saying. Clear? This is what they are saying. Next one. In the case of eukaryotes, not just one. In one chromosome, many origins of replication are there, but anyhow this point was not given in NCERT. Next one is an important, very, very important previous NEET examination question also. Helicase enzyme. Sorry. Helicase enzyme, it is also called as unwindase. Here, I will directly tell you the two important things. One question is related to DNA replication. One is related to transcription. Anyhow, in the transcription, in the next revision video also I will tell you this point. Remember this. During DNA replication, the two parental DNA strands should be separated. Yes or no? Now, who will separate these parental DNA strands in the DNA replication? It is the helicase or unwindase enzyme. Very good. During transcription also, both the parental DNA strands are separated. But who will separate there? This is the previous NEET examination question. During transcription, who will separate both the parental DNA strands? It is the RNA polymerase itself. This is very important. It is the RNA polymerase itself that is separating both the DNA strands in the transcription. But during DNA replication, there is a separate enzyme called as helicase that is coming for separation of both the DNA strands. This is very important. Clear? Next one, regarding the single strand binding proteins, not mentioned in NCERT, but better try to remember the logic. Helicase enzyme is separating both the DNA strands, but both the DNA strands are complementary, so there are chances that again they can come and attach. If they come and attach, there is no use in separation, right? So once helicase is separating both the strands, there are some proteins called single strand binding proteins. After separation, single strands are produced, right? Immediately they will come and attach to this, so that they will maintain in single strand, so that they will not attach again. So that next enzymes will come and they will start the synthesis of DNA. This is what the advantage of this, but not mentioned in NCERT. Next one, NTPs and DNTPs, very important. Even though it is the DNA synthesis, anyhow for DNA synthesis, deoxyribonucleoside triphosphates are compulsory. But even though they are compulsory, now even nucleoside triphosphates means ribose sugar containing nucleoside triphosphates are also coming, because there is a small RNA primer that is produced by primase enzyme. Primase enzyme will produce small RNA primer. For small RNA primer, these NTPs are required. Anyhow, later on they will be removed, but for initial primer synthesis they are required. So that's why NTPs are also required. Anyhow, DNA for DNA synthesis, DNTPs are required. And very important point is, these nucleotides will play a dual role. What are these dual roles? They are acting as substrates and they are also acting as energy sources. So when they have given this sentence, within bracket they go, in the nucleoside triphosphate, 3 phosphates are there. Out of these 3 phosphates, last 2 phosphates, terminal 2 phosphates, very clearly mentioned in NCERT. Terminal 2 phosphates are linked with high energy phosphoanhydride bonds. That's why terminal 2 phosphates are called high energy phosphates. Last 2 phosphates are high energy phosphates. Very important for examination, remember this. Next one is primase enzyme, not very clearly mentioned in NCERT, but please remember in the NCERT they have given one sentence. What is that sentence? DNA polymerases cannot initiate DNA replication on their own. That's what they give. That's it. DNA polymerases cannot begin DNA replication on their own. That means they cannot start on their own. That means someone has to synthesize a piece of DNA or RNA so that they can extend. So please understand this. So indirectly they are talking about this. Since DNA polymerase cannot start replication on its own, it requires an enzyme that is called as a primase enzyme. So primase enzyme will come first, and it use NTPs, means ribose sugar containing nucleotides, and which will produce a short primer. It will produce a short RNA primer. It will produce short RNA primer. What is the advantage of primer? Primer will provide a free 3' OH group, so that in the next DNA polymerases will come. DNA polymerases will come and they will continue this primer. And once it is continuing primer, role of primer is completed and then primer will be removed. So that's why this primase enzyme is very, very important. So please remember this. Okay, next one. So since helicase enzyme is keep on separating both the strands, when it is keep on separating both the strands, here one helicase is going on this direction, other is going on this direction, because replication is occurring on bidirectional, on both the sides it is happening. So now what happens, this separation will create some supercoils in front of replication fork, and someone has to release these supercoils. If these supercoils are not released, then what happens? Now this further opening cannot happen. So there are separate enzymes for this particular purpose. And please remember, in the case of prokaryotes it is called as DNA gyrase. In the case of eukaryotes it is called as topoisomerase. But anyhow, topoisomerase you don't need to remember, because we don't want eukaryote information, but prokaryote information is important. So DNA gyrase function you please remember. Helicase helps in separation of both the strands, and gyrase helps in releasing or removing supercoils that are formed just in front of replication fork. At this opening you are observing Y-shaped fork, and this is called as a replication fork. At the replication fork, at the small portion of this is a small portion where DNA replication is continuing like this, very clearly mentioned in NCERT also. Next we are talking about very important enzyme. They are called as DNA polymerases. For DNA synthesis definitely DNA polymerases are required, but very clearly they have mentioned just now also I told you. So what is that? DNA polymerases cannot initiate DNA replication on their own. That's why primase will come first and it will produce a small primer. And this primer will be continued further by mainly DNA polymerase 3. This is the major replicating enzyme. In the NCERT they have given the speed of this one also. Approximately 2000 nucleotides will be base pairs. Approximately 2000 base pairs are synthesized per second. In 1 second, approximately 2000 base pairs are synthesized by this enzyme. That is the speed. You can understand at what speed actually it is able to synthesize. In this way it is keep on doing. And once that primer is extended by this enzyme, that primer function is over. Now this DNA polymerase 1 will come and it will remove that RNA primer, because RNA should not be there finally. So that RNA will be removed. And then slowly it will add some nucleotides of DNA. Okay, so in this way ultimately at the end you will get entirely DNA only. Okay, this is what. Anyhow, this number 2 is involving in repair enzyme, but the major point is in the NCERT they have not given 1, 2, 3. They have given only one enzyme name, and that enzyme is having 2000 base pairs per second speed. No, that is actually number 3. That is actually number 3. Remember this. And then leading and lagging strands. During this process of synthesis, what happens is, here the original parental strands, blue lines are separated here. Yes or no? And each one is acting as template. And please remember, always synthesis is from 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime. In this way it is happening. So in this process, what happens is, one strand is synthesized always ahead of other strand. The one that is synthesized always ahead is called leading strand. And whereas the other one that is synthesized slowly and it is not ahead of this and actually it is lagging is called as lagging strand. Please remember this. Next one. The one that is the leading strand is always synthesized continuously. That's why leading strand is continuous strand. But the lagging strand is actually synthesized in the form of fragments. What are these fragments are called? Okazaki fragments. And that's why it is the discontinuous. One is continuous, other one is discontinuous. That's why DNA replication is called as semi-discontinuous. In the previous experiments, we proved that it is semi-conservative. So DNA replication is semi-conservative and semi-discontinuous. In this way it is happening. And please remember, leading or continuous strand is synthesized towards the replication fork, whereas Okazaki fragments are synthesized away from the replication fork. And with respect to fragments, if you see, the fragment that is present nearer to replication fork is latestly synthesized, whereas the fragment that is present far away from replication fork is the oldest one. So in this way, this also you need to remember here. And for the leading or continuous strand, only one RNA primer is there. But for each Okazaki fragment, there is a separate RNA primer is there. Anyhow, later on it will be removed. This is what the thing is. And finally, so this is how this synthesis is taking place. Next one, DNA ligase. What is the function of DNA ligase? So wherever the nicks are present, that means suppose here, so this RNA will be removed and replaced by DNA polymerase 1. And finally, there is a lack of one bond. And between the Okazaki fragments also there is no bond. And wherever there is no phosphodiester bond and these nucleotides are there, but bond is not there. So those will be linked or sealed or ligated, sealed by ligase enzyme. So this is the function of this ligase enzyme. It helps in the ligation or sealing of these particular gaps. Okay, that's what they are technically talking. So in this way, there are different enzymes. They are having different roles. In this way, in the NCERT, they are talking about only the machinery one. Finally, it is about the divalent cation like Mg+2 or Mn+2. They are very important for the functioning of DNA polymerase enzymes. Clear? This is what they are talking. And that too, the DNA polymerases that we are studying here are DNA dependent DNA polymerase, because they will use DNA as template for the synthesis of DNA. DNA template for synthesis of DNA. That's why DNA dependent, depending on DNA template, they are synthesizing DNA. That's why they are called DNA dependent DNA polymerase. This name also they have mentioned in NCERT. So please remember this. So, and these DNA polymerases require Mg+2 or Mn+2 for their activity. This point was not mentioned in NCERT. Clear? And bidirectional DNA replication. So DNA replication in the E. coli, it is happening in bidirectional. Even in human also it is bidirectional. That means this is the replication fork. So this side also DNA replication is occurring. This side also DNA replication is occurring. That means both the sides DNA replication is occurring. But the point you need to notice is, since that 5 prime to 3 prime direction has to be maintained. See here. If on this side, upper side continuous, bottom side discontinuous, on the other side it will be reverse. Here bottom side continuous, upper side discontinuous will be happening. So in this way, this DNA replication is occurring bidirectionally. But anyhow, with more clarity, with better explanation, I have given in the regular lectures. So you can go through this particular information for better understanding. Anyhow, with a sequential flow also I have very clearly explained, so that you can never ever forget this particular information. Clear? You can easily remember also. You don't need to work too hard for this one. So okay, anyhow, since it is a revision lecture, this much is enough here. And finally, in the paragraph they are saying that, so in the case of eukaryotes, during the S phase, this DNA replication is taking place. So in the S phase, DNA replication occurs, and see, because of this, chromosomal duplication occurs in the S phase. In the S phase, DNA replication, chromosomal duplication occurs very clearly. I gave this explanation in the cell cycle chapter. Go through that. And this replicated chromosomes, that means sister chromatids, are formed in the S phase. And now they will be distributed in the karyokinesis process into 2 nuclei. And it will be followed by the cytokinesis, so that now they will go into the daughter cells. This is the general process. But if DNA replication occurred in the S phase, but that cytokinesis does not occur, then what happens? In the single cell, Now the double number will be happening, so that now what happens, it will result in the polyploids formation. So this is also a type of chromosomal anomaly. Anomaly means abnormality. So in this also can happen. So in this way, if cytokinesis does not occur after completion of DNA replication, that will result into polyploids condition. That's it. With this up to the DNA replication, the first part of this MBA revision is completed. Clear? Before concluding, once again I am telling you, revision lectures are available both on Live Prep app and also on YouTube channel, but test series can be given only through the Live Prep app. That's it. Clear? So in the next lecture we will go with the remaining part of the molecular basis of inheritance. Thank you.